vessel away. Well, yeah, without going up.
While we're planning, can we get the beacon fix?
Yeah.
Hey,
Josh? Do you mind?
I think the idea is super then get the lander, but it would be nice to economize travel time.
Or did we already get it?
It should already be up there.
Oh, is it? Okay.
It's just south.
Oh, you up?
Should be, you zoom out.
Oh, mini lander 2?
Mini lander, I just dive 2.
Yeah, it's up there.
Where is it?
I think.
Here, let me just, 1 second.
Yep.
Well, I can't
Standing by.
virtually invest with it anyways. Waits once it gets on its way.
Mm-hmm. I just want to make sure we're not headed in radically the wrong direction if it happens to be on the north side of the scarp.
It's southwest of us.
Oh, everything's southwest of us. That's where all the action is. But do we? Is it?
Yeah, it's all the way down here. You see this?
Oh, great. Okay, then let's proceed to active creep, and we'll deal with the lander afterwards.
Did you show me that? I missed it.
Okay, and then we're live now.
Thank you.
Yeah.
Great.
And audio works.
Oh, the yellow.
Okay.
It's this one, right?
Thank you.
What? The lander? No, it's a blue one. So BBC lander.
Fish.
Yeah.
Oh, it's way yellow.
So we landed it in that general.
Welcome to those joining our dive online. This is SuBastian Dive 0566 on the Pinguinos vent field on the Western Galapagos Spreading Center. We reached bottom less than an hour ago. You're in the control room with the Dawn Watch Donner Party. That's DAWNER. Haley, Rachel, and Jacob. We've collected a dandelion siphonophore, which I can talk a little bit more about shortly. Now we're headed to an active venting site, looking for new active venting sites along the way. We're going to do some filter samples using the super sampler there. And after that, we'll do some lander ops. Stay tuned.
So, hey, Josh. Hey, Josh.
Yep.
Did you hear me in your headphones?
No.
Okay. Would you mind switching this to the SITCAM so we can fix the autofocus?
Yeah.
Would you mind switching the autofocus setting that?
So the animal at 949, which we sampled, looks superficially like a sea anemone. It is a cnidarian, belongs to the jelly phylum, but it's a really peculiar cnidarian. It's not an anemone, it's actually a siphonophore that lives on the bottom, a benthic siphonophore, which is probably new to those of you who are familiar with siphonophores. We usually see them in the midwater. So the dandelion siphonophore, which tends to show up around vents.
Yep, backwards.
Is sort of an example of a lineage that evolved first to live in the midwater, and then evolved to live on the bottom again.
There. Should switch it to manual. Alright, now let me make sure it changes. Yep. That's correct. Cool. Thanks, man.
Oh, there must be something else at 949 that I'm not getting. Or, well, I can't watch the recorded footage, but we did see a flytrap anemone shortly before the dandelion siphonophore. So I'll leave it to the two of you to tease out which is which. The dandelion siphonophore looks like a dandelion. What do you think, sir, ETA? Something like 10 minutes?
Until we get to active 3.
Yeah.
I was thinking a little ways because we still have to go. Yeah, that'll be about.
Okay.
The boat's gonna be the speed.
Sure.
We go at.
So it's about 100 meters.
Mm-hmm.
It did.
Oh, yeah.
Yeah. It did. It's like a little disco ball. Gorgeous. It's still in there. You wanna
What's like a disco ball?
a little team port.
Oh, it is not still in there.
Yeah. Where is it now?
It's in the freezer.
Oh. It is. It will no longer light up for
It's
you.
okay. Yes, I'm all right.
Genetically, it's still a little disco ball.
That's all that matters. On the inside.
It lost its shimmer.
It's okay. That was the one point which I was.
It was pretty cool.
Hopefully, we'll find more. Though, okay, who's that?
What is this little critter?
Yeah, let's let me know when I can zoom on that.
Let me get you closer in the center.
Oh, it's a
It's a fish.
Yeah, it's just at a weird angle.
I think it's
Blinded it.
okay. That's great.
And
Oh,
you want to just.
is it just a Zoris?
Iris down. I don't think it's a Zoris.
It is.
I don't know. I kind of don't think so.
It's a little long.
I don't think so at all. No, we haven't seen this guy yet. Whew.
Why?
Not a snipe eel either, but it's kind of shaped like one. Just mark fish.
Fish, just fish.
There it goes.
Weird fish.
Back.
Oh,
Back.
That's it. You go ahead and take the camera.
yeah.
It's gonna just run.
All right. I'm gonna pull in on it for a second. We'll take an observation and. Huh. Look at that. Look at that. My autofocus is.
Eyes are like kind of on the top of his head.
Makes sense.
Long fish.
Hmm.
It's quite weird.
All right. We got the observation.
We have observed this fish.
Okay. Back in autofocus.
Lights are camera.
Except not. Now we're in autofocus. You brought the camera back up for me?
Yeah.
Thank you. All right, let's keep cruising.
Let's get these boats in that apparatus.
It's back.
It's back. Once more.
This
Well-
patch up here looks
Hello again.
awfully good for sediment.
Playing with fire, that fish.
Right now. It's really a spotlight.
Right here? Further back, or is this the patch or right here?
I'm sorry?
Is this the patch or further behind us?
Yeah, I think this looks
Hmm.
okay. So maybe up here.
Very shallow. Good question.
Yeah. Drop a marker.
Drop a marker.
Yes, please. I think if you can check yesterday, I think this is going to be sediment 3.
Oh.
We left off at 2.
Oh, okay. I thought you meant this was a previous
It's really small.
site. Sediment 2. That's correct. Sediment 3.
Sediment 3. So just
Mhm.
really small patch of distal sediment for fish course. So just a marker here and we can keep going. Thank you.
What do you want to call this?
Sediment 3?
7.
You just want to call it distal sediment for push coring?
Yes. But just say really small patch.
Very small patch.
Very small patch.
It's very shallow.
But I'm getting desperate. So we're gonna mark it. We found a little mound. One of the first dives.
A
and
mound
got
of-
really good cores from it.
Hmm.
There were like a couple small mounds that worked really well.
And when you say mound, do you mean a mound of basalt that had sediment on it, or a mound like a hill
mound
of sediment?
Yeah, amongst the basalt. Had some volcanic glass in it.
Oh,
It was very sandy.
cool. Did you see it sparkle?
Hmm?
Did you see it sparkle?
Not from the ROV image, but when we sampled it, some pieces. Just something to look out for.
Mm-hmm. Thanks. Just out of curiosity, Alex, are autofocus and auto iris linked? Like when you engage autofocus? No. Okay.
You can, the status of the autos for those settings are just in the bottom right corner of the camera.
Uh-huh. Okay. Oh, I see the iris row now.
Usually we can't roll the iris all the way open. We
Mm.
just try to darken it bigger.
Mm-hmm.
I sort of shutter speed. Shutter speed's usually 150, 150.
Oh, interesting. You don't like to darken it with the iris.
No, because I guess from what I understand about photography,
Uh-huh.
it helps like for post processing. Like the sensor gets as much like actual all the colors and light and whatnot. You can correct for things. Whereas if it's dark, it's hard to brighten it.
Certainly.
Yeah.
Uh-huh. Sure, if you're recording raw, that makes sense.
But like I said, I'm definitely
Yeah.
so.
Uh-huh. And the advantage, of course, is that as you stop down the iris, your depth of field increases. So you get more stuff in focus. The focus becomes more forgiving.
Oh, got it.
Your iris down.
That's right.
Oh yeah, another long skinny guy in the background. So it's the same fish.
So your depth of field on an ROV isn't very good just because the lighting doesn't penetrate.
Must be fish too, and there's this,
It's
that's
Now that I think about it.
But it
Yeah.
makes sense, though.
Well, it was pretty much our universal practice for our midwater work, but we were all manual focus, and you're in the midwater. So if you want a shot that's steadily in focus, you take advantage of that. But I see how Propenthic
so
works.
when we do midwater stuff, we'll typically leave it manual focus also.
Mm-hmm.
And then only until we think we're confident that the autofocus could possibly track an object, we go into auto.
Mm-hmm.
Do you want to call this a sediment patch or no?
It's too rocky.
Rocky. Rocky Poke. What is this? Jacob, what is this little critter?
That looks like a sponge to me.
This looks like a sponge to Jacob.
It's almost certainly a sponge.
That's,
But I also don't.
this is where it's sea-logged and it gets real.
I don't have ice control, my hand control, so I use iris because I don't.
Mm.
It's like,
I can really see why you want to lower the ISO. I mean, it makes for a nice blow up frame grab. Less noisy. Let's keep going.
That's some nice sediment. Maybe.
you just told the internet that, man.
So what I have here on the objective list, Rachel, is that we want to super from the same place the IGTs were,
Yep.
the little stub,
Yep.
and it says here is our innovator.
Yeah,
Is that
that's just,
correct?
yeah.
Okay. That's great.
Yeah.
And then are we also
No, I'm just kidding. I can only hear you because I knew what you said.
Do you want to grab what's left of the chimney
Yes,
and
put it in
orient
the
it?
coral quiver.
Cool.
Yeah.
This time of night, it's all volcano.
No, yeah, no one heard that.
Morning, everyone, and thanks for joining us. This is our second dive on the Pinguinos vent field. Right now we're headed back to an active venting site that we took samples on yesterday to take some more of a different kind of sample. We're looking for sediment that we can core along the way, but as you can see, this is a pretty rocky area and it's hard to come by. A lot of those little stalked sponges, clitorizids maybe. I don't know. Have you ever picked those, the little stalked sponges? Have you ever looked at the stalks?
I mean, probably not.
They look like optic fibers.
Yeah.
Yeah.
We pulled some sponges out of the sediment that had like roots almost.
Yep,
Like
that's exactly
that were,
what-
like,
yeah.
4 or 5 feet long.
Whew.
We only got about 3 feet.
Okay.
Temperature normally.
Still pretty good.
Probably about- But yeah, like spiral them up like spaghetti and biwalks or something like that. Wasn't very sandy seabed.
Mm-hmm.
I think there might be a vent nearby because there's kind of a big temperature anomaly.
Yeah, there's
Mm.
stuff right here too.
Okay.
Let's see. Which way is the current going? It's pretty still.
She ran about 25 meters.
Could be that one. Yeah. Let me know if we can turn around.
At least.
Where do you think the smoke's coming from?
I can stay still if you want to try to.
We're about, well, we know that we're about 35 meters from active 3.
I don't think we would see this
Yeah.
kind of anomaly
Yeah.
that far out.
Yeah, it is pretty toasty.
Set right here. Let's see if we can't see where the current's coming from.
I think it's coming from the right.
We just have to make sure it's not a prop wash.
So right now, the pressure should be pushing back towards us.
Okay.
Like, towards the back.
They're pushing astern.
Yep.
Mhm.
That's the looks good. So can you tell which way the current's going, but? Yeah. It does look quite still.
Hmm. Do you think it would help to gain a little altitude?
Maybe, yeah.
All right. Yeah. Pop up a couple meters, maybe.
Coming up.
Maybe we take a little. Yeah, that's probably well, and just take a little look to the left and right.
Oh,
Whoop.
that ought- sorry, that was a good-
Auto def.
Yeah, that was nice. Forward, just a little bit.
Well. Don't see any shimmers. Oh, what's going on? No. Nope. You sorry?
Just open. I guess we're left now.
There isn't anything directly below us, is there?
Is that the little, what?
Where?
Straight ahead.
Straight ahead?
Yeah.
Can I tilt the, or you can drop down? Okay. Tilting down. Where, in the side cam?
No, the one the super near chimney.
Oh, yeah, the super near chimney is not a chimney. It's 10 meters off the chimney. The chimney is active 3.
Active 3 is over here.
Yeah. It's active 3 is almost 40 meters to the south. It's rising?
Yep.
Yeah. Yep. Don't
So we're near something.
near something. I mean, we're reading in the hundredths of degrees. Like,
I mean,
I trust your instinct here.
that's.
Oh, it's going up.
Yeah.
Are we on top of it?
It looks like the current might be coming
From-
over
Yeah.
a little bit,
Yes.
but that would be kind of going. So that'd be going towards.
I know it's going away from our target endpoint or
It is.
target waypoint.
Would be going towards that.
Uh-huh.
Maybe if you want to just drop a marker and
Yeah.
just say temperature anomaly.
That seems why it's so tempting to look at that embankment. But yeah, let's drop a marker and call it it's a long. You can call it temp anomaly 1. And that's an excursion to 2.60 Celsius.
2.62.
2.62.
Or during that, take. I'm gonna put a half turn in the other direction. So
Sure.
turn the other way, so
Go ahead and take it out.
well, yeah.
Yeah, I know that's not my call to make.
It's not. Yeah, it's not. Well, I mean, it's not a big deal. It's just going to be turning that thing.
Sure.
It's just not do it.
Mm-hmm.
We're kind of off the scene, but-
That's us. That's our own.
that's-
Yeah.
that's wrong. Wrong. So just literally just about all the way around, but then
Yeah.
it's a turn, half a turn in the other way. It's actually better. You know,
Uh huh. Well, it's half a turn in the direction we're gonna go. Yeah.
Josh.
All right. Whenever you're ready, we can proceed to active 3.
Josh. Yeah, something wrong. What's up?
Our delta, our depth dropped out.
Oh.
They want all the information from Delta's now at 17.
Hmm.
Depth invalid.
How safe is it to fly without those data?
No.
Delta's troublesome.
Yeah, we can put our depth. So we can
Why is there? So depth is.
see what our sensor.
I think there's a harp sponge in the frame and left.
Arrow down? Is there any reason why I can't use it? So you or
Do on S depth is going up.
Okay.
I don't know why arrow depth should be the same. Okay.
That works.
Good.
Okay. We're in business.
So you have a separate arrow? Or is it?
We've got, yeah, I think there's 2 separate. Yeah, I think so. But is the Paro feeding the INS?
INS could be on.
I turn the power on over here, so the Veilport IPS
Yeah.
that gets put into the Sprint system, you turn on in Sprint, which is why I'm almost wondering what that Paro, I'm not too sure what that Paro is, if we have another Paro sensor. I think we do.
Yes, we do.
So next time we're on deck, we can take it off and see if something's up with it.
Now running INS then?
Yeah.
I don't know
Yeah, a little sponge party.
See if it does it again, right?
Yeah. I didn't see any. Didn't see any bivalve beds
When
either.
we come up, we need to check that. CTD depth.
Yeah, it's climbing towards the bottom of the embankment. Well, that's the direction we're headed. We're headed towards some known vents. Beautiful sponges. Yeah, yeah. I think you can label that anemone. I think that's what it is. Could be some kind of cup coral. Can we stop down or something? Okay. Does that get me manual iris? No.
Yeah.
Yeah, there it is. There it is. Oh, it's always manual. Yeah. All right. Let's keep going. Oh. I mean, I think we can keep going. I don't think we need to set down. Yeah. Thanks.
What's that up ahead to the left of the sponge? Is that just
The
another
long
movie?
thing?
Yeah.
I can't tell from here. That right there.
Oh,
Yeah. We can fly on it, or if you get really steady, I can zoom it. Sure. It looks like an anemone.
Big ol' anemone.
Don't know what kind. But it looks similar to the other one. Possibly a different color morph. This is a nice little rock, though.
Nice.
Yeah. Cool. We got the observation.
It is observed.
Let's keep going, please. So observant. Harp sponges on the upper left. We're not gonna fly on that though.
Anybody has any idea how old these pillars are?
The spreading center's 12.5 million years old. So somewhere between 0 and 12.5 million.
That's the kind of error bar I like.
Yeah,
We're back up at 2.62.
Supplies are not covered with more sets.
that's interesting.
Yeah, so they're probably fairly young,
That's what I thought.
given the lack of sedimentation.
Yeah.
Is that something that that like your team can get from the mineralogy? Can you date these basalts?
You need zircons.
Mm.
We date the sediment cores.
Mhm.
We use C14 dating. We don't do that on the rock samples.
Yeah. You would need ash in order to date the rock
Hmm.
to get precise dating from zircons.
If we get more push cores.
Yeah.
But active freeze, so we're falling there, still, but active freeze.
But you're right, it is relatively young. because
Then let's hang 90 degrees
How
before
young cell?
Barely any
and
sediment.
head for active 3.
The less sediment.
Thanks for the update.
is-
I know, so how young? Not 12.5 million.
I don't know.
Yeah.
I've seen lava pillow form underwater. After 6 months, they look like
We're also like kind of in the what is it? Near an OMZ.
Yeah, I was
So
gonna.
we're in a gyre, so there's not going to be a lot of sedimentation.
So how quickly it kind of mucks up is going to depend on the organic load in the area.
Yeah.
But
Yeah, what's the oligotrophic sedimentation rate
right. So 0.3 centimeters per 100 years. It's very low.
Okay. We are just about there.
Whatever, 0.3 is in my mind. I don't know if you guys are talking.
0.3 is what you're wanting.
It's on my mind.
1 millimeter per 1,000 years.
Okay, I was off by an order of magnitude. It's all right.
This is what the Pinguinos
I see a chimney.
stand by. Small clay particles deposited to the deep ocean about 1 millimeter per 1,000 years.
Okay.
So, if I recall, this feature, catch me on this if I'm remembering the wrong one, but I recall this feature is the one with the active chimneys in the gap,
Yep.
right? So there's two mostly capped mounds, and then there's like a little canyon in the middle, and that's where our active venting is. All right.
Active 3 is the one that had like 3
Yeah, the-
oxide
Right,
spires.
right. And I'm actually small height to chimney top structure, white sulfur. I didn't record our heading, so hopefully we end up on the right side. This is, I believe, the right side. So,
It's very pretty.
yeah, keep coming around it. I think that we want to be like over here.
That's rough.
I think. Oh.
I see shimmering fluid.
Yep.
But I don't think that's the.
Let's see if our friend the crab is still here.
This up behind me too.
Pumped.
Sorry. No, I should watch you. You can come up so you can see some of the other stuff.
Whoa, it's an Aegina. What are you doing down here? Also known as a lemon jelly,
Lemon
for those of you
jelly.
listening online. Aegina citria.
Not limoncello. I wouldn't pick it up because it's stupid finicky. I might just.
Are we switching
Hmm.
up to longer?
Say again?
Now longer one.
If you don't mind.
Yeah, of course. I just like, sure. Didn't want to take over you. Are you fine?
No way.
I believe it's the structure on the right, but yeah, this is kind of treacherous territory, isn't it?
There's a third one.
Oh, it looks like, I mean, if we trust the tracking, we're a good 5 meters east of where we want to be. We want to lateral to starboard.
Yeah. Right, so. We've got stuff to the other too. Probably. Yeah. It's also this.
That's the one we just passed.
Oh, it's much slower than I realized.
So that's nice watching on your screen.
Yeah.
That's the one we can put back.
Yep. Around all here, so you get a perspective of what.
These things will pick up on sonar, right? They're not too small.
Yeah,
But it's only facing one way.
It's like a super pretty slow update. So
Mhm.
I'm changing heading a lot.
Yeah, yeah. I see what you mean.
It's just the two. It's
Super
towers.
Oh, they're probably almost.
Yeah, I think this is the feature we're targeting. We also never got a height for this mound, so that would be worth measuring at some point. I didn't get a height for this mound. It's not in the marker notes. It's this feature, but we're looking for that canyon. I think you have to come around one side or
Right,
the other.
and go down. Hand down.
Down and to the right.
Yep.
Okay. Do you think
That's it,
that's
that's it.
the inactive,
That
and then
is-
the
yes,
orifice? That is
Yeah.
where we want to sample.
That is correct.
Maybe we need to stop down, but yeah, that's it. Bingo.
Bingo, bingo. I told you, never forget Jimmy. Can we power on super?
I just need a quick note on the checklist.
There's your crab.
Yes.
I'm gonna check out the
Checklist. And there it is.
Pretty hard.
Oh yeah.
Well, I don't know if
Here, you want to click forward to?
looks okay.
It's okay,
Will Chim returns.
Oh, wait, this is the one where we're at.
Yep. That's exactly where we want to be.
This is flashbacks.
Mm-hmm.
Got it.
Deja.
Let me let that clear out a little bit. Tighten it
Fire up super.
Thank
Super
you.
power.
Super power.
Thank you.
And then if you're going to pick up the lander on your watch, you can just keep an eye out for something.
Roger. Is that what you wrote down?
You know, I
No,
marked
looks
this
like Rachel.
yesterday as a possible corner spot that no one has tried.
Try an active one.
That's close to F, so east of you.
Okay.
And then this is what we just marked. I think it's a pretty low
I
chance.
was just concerned.
Okay,
about
to give
Mhm.
that to
Just-
Okay.
geology.
Do you need to be woken for coring activities? What's the plan?
Let it sit down.
Sounds like it's not going to happen on this watch, right?
That seems unlikely.
Yeah, that's
It'll probably happen post lander work.
I
Yeah.
think Amy's been getting up for the next watch.
Mm-hmm.
So someone from my team should be up for.
Okay.
Some of that is.
Sounds
They need
good.
to text somebody.
I'll let the next watch lead know. Thanks, Caitlin. Much more.
The what?
The delta, yellow change red. For which? So, 3 times change red, so. Well, it's when you do that depth reading, I think, or to 0. Remember that change, the code.
Or whatever, it drops out or something.
So what are we doing?
What are we doing? We are taking a super sample. So we're gonna I think we're gonna do a near orifice super sample first.
Yes,
correct?
so,
Rich, we're on the
pull
show.
out the super wand and the temperature wand, and I'm aiming for fluid that's around 40 Celsius.
Work on the porch a little bit. Just crushed a crab under our nose. See that? Probably shouldn't show them science, but that hose dragged over the rock. It's like penguins crab down. Can we
We
save it? Great. Alright, let's grab the stuff. I can grab the super if you want to grab the temp.
We are going to sample.
Yeah, but I figured we can get them both out.
Yes,
So we got those.
we sample the yellow, right?
Yeah, we're gonna be up there, which is just ahead of us, but here, this is a good spot to have the porch out.
We'll use temperature first, find the 40-degree temperature mark.
Yep.
Once I find it, then we turn on the super.
Yep.
So,
Is that far enough?
that's fine. Tell me what you get first. Once you get over there.
You'll also pull the super out and have it ready. Your sip food.
Correct.
But the dredge is also snagged on the port right, so I'm not sure it matters. A little more quick. That's good. Alright, come up. Actually,
So we want to sample the flow in a spot that's about 40 Celsius. Just as a reminder.
remember where we landed on this.
Was it smoking this much yesterday?
It was more shimmery yesterday.
Could be the light.
That is smoke, no?
Yeah.
No, we can't see on the set. Well, we'll see where 40 is. I can also come in on the. I've also set up a little bit to the left for yesterday, so let me know if that causes you problems. Just a little bit.
You see the little nubbin, Rachel?
Yeah.
They can grab.
Yeah.
Do you think that'll do?
Yeah, that'll be fine. It's the same prop from yesterday.
What's the temperature?
9, 10,
12.
18, 22, 26, 31, 35, 37, 35. Looks like you can go, oh, there you go.
Nailed it.
It's 41 and a half, 38.
40 is what we're looking for.
Yeah, we're looking
Yeah.
for
Back
40.
down now.
The current was waving back and forth here yesterday.
Yeah. Easy move.
Yeah, come on.
You're a little off center. Yeah. That's too hot.
There, just 2-1. So you want to come up in the column?
Roger. Same line.
Keep coming.
How's that?
44, 42.
Might come a little bit to the right, I think, because we're going to have to get room for the nozzle as well.
A lot of happy albanellids here.
So we can reach it.
It's not. What is it now?
Plus 20. I think we're past towards away from us.
6, 29, 34,
There it is.
44.
Hmm.
Lock.
Still 1.
It's one.
Little too warm.
47.
What is it?
I think this looks good. All right,
See if we can reach it.
I am going to begin popping.
Alright, thank you. Let me see if we can reach it.
47.
Oh, no, wait, no, not yet, because we have to move the super.
Yeah, we don't have the nozzle there yet.
Yes.
Hold on.
Hold on. A little trigger happy.
That hose is pretty long. Yeah, it's budging down kind of low.
So we've set up on the same active chimney that we took isobaric gas tight chemistry samples from yesterday. And now we're deploying the super sampler, which is going to pump some of that plume water onto a filter, and we'll preserve it with an RNA DNA preservative. The tool on the right is measuring the plume temperature, and the tool on the left is just an intake nozzle for the sampler. Now, let's not get too much lower because we don't want to suck really hot water into the super. It makes Roxy nervous.
It's not even.
It's even?
No, I'll try to freeze it.
You want me to freeze it?
I'll try one more freeze if that doesn't work. Okay, yeah, we did it. Just a second. Back. Right there. Come up.
Yep.
Locked?
Just touching it with it.
Okay, and begin pumping now.
It's gonna lower it because
That's
it's pulling in
fine.
ambient water with it.
Yeah.
And it's like right at the inlet.
Rachel, is this going to pump onto filter 1?
Yes, this is going to filter 1.
I didn't hear what you were saying.
complete.
Oh, they're back.
Grabs your back.
Oh my.
Okay. Pumping. Sample 1. Starting now.
What's the official stamp on that, Haley?
The
Any guessing?
official stamp when we started purging was 1056.48.
Thank you. I'm gonna grab my water bottle from the labs. Anybody need anything while I'm up?
Turn it off. I put it in the board.
Okay.
So it's all turn it off.
See the text here.
1056.48. What I purged.
Oh.
Was 1059.
How did she come there and start this down? Yeah, how did you go first?
It's one for sure.
If they can't help us somewhere upstairs, I probably have a contact with them.
Same thing. So, hopefully, it's up to
Yeah, that'd be great.
Latitude
No, they were
is
stuck to these.
2.099.
No way.
428.
No way.
Okay.
Longitude is negative 1.
I swear to God. I was like, what the hell is this thing? Knocked them off
Okay.
there onto the ground.
Okay.
Not mine.
Yeah, they were on the ground first. Get rid of them.
That's it, man. You snack on them, all that.
You can just put it in the empty office.
Can't stop the ship.
There may be more than four.
Bears? Which is an hour ago.
I've been walked off this boat. Still, boxes are done.
Go. There's one under here for all that stuff. I don't see it anymore. I think the rest is because it has the lock-in writing on it.
Fortunate. Won't be able to stop the thing over. Oh, I didn't have to take this. All that was.
Oh, look at that cute little crab in the foreground.
Yeah.
Mostly fuzz.
Just drifting away from us.
What's drifting away?
The town.
Yeah.
It's all right.
the man.
Yeah, that's a nice frame, actually. Do you mind if I back out a smidge? Yeah, thanks for setting this up. Oh, I got the
So cute.
tools above.
Nice little worm.
Yeah, I want to see how much of a frame I can get without the nozzle in there. The answer is not much, but it's nice to look at.
Let's see.
Can you take
Flouting.
an observation of that?
Yeah,
Just
I
picture
got it.
Flouting Dan's rule of thirds.
Run the camera.
Because I was like, you might be going.
Where is it?
If we're looking up,
We're
if I ran the camera out at like,
we're panned right, so you rack out.
put them,
See what you're
I
saying.
mean, you have to.
And the one you guys shot.
See what you're saying. Oh, sorry. Go ahead. Take it.
Oh, it's better just do it. I just wanted to see the worm. I was hoping they were going to get a fight. But they go, we have it.
Here. Only one hand touch.
I don't want your gummy bears. No, I know at least two hands touched those because the person who put them there, and then when Doug knocked them off.
No,
Not
it's
you.
new.
Oh, is it?
Yeah.
So just Josh's hands.
It's one hand.
You guys make me nervous. Can you turn off the lowers? It'll go a little less bright. Is that what it was?
Your hands open.
Nice. And
I don't keep shadows.
I'm gonna close up a little bit. Did you say you cut the lowers?
Where did these come from, George? I did. I can turn back on if you want.
No, I think I think it's good with them off. Hmm. Do you have independent left-right control on the lights?
No.
No. Okay.
Some of the lights.
Well, one of the lights is only on one side, so
Which is that?
inner cathodics.
That's wide beam, right?
Sort of. I mean, just kind of pointed near field edge.
It's near field and the lights on the starboard side.
You want to try it?
Yeah, let's try it. Well, let's try cutting the others. Go for some dramatic side lighting. Ooh. Oh,
I mean, from the
that's
left side, it's right on the
Well, a little
arm, so
dark,
it's not
yeah.
gonna do anything.
Just a wee bit. It's a little too much drama.
Yeah, I should go back. No, it's right above the arm, so it's not.
got it. It's not gonna get there.
I
Wow.
brought one.
It's a bit dim.
Triggers are on.
How's our timing on the?
We started pumping at 10:59, so
So we have a couple more minutes. 3 more minutes.
Why are you smiling? Why is it comfortable when someone brings you food and then looks at you smiling? Nothing wrong with that. You have to share very much with it.
Oh, the current just changed.
Is it possible to switch out the temperature wand with the pH probe and not disturb the super wand?
I'll look at the,
I think you can do it.
Are they touching each other?
We have to come
Can-
back. The lowers real quick.
Are you gonna use the temperature probe again?
We will, yeah, for the next sample, which will be straight up from here. So would you rather log continuously with the temperature wand and then go back after sampling and put the pH probe there, or switch back and forth, which is easier?
Are you telling to us?
Yes.
If we're gonna use it again, I'm just gonna lay it on the basket on the
Feel
box
free to back us out.
right now, instead
Okay.
of putting it back in the holster,
Okay.
which takes a little bit more.
Hmm.
I'm gonna just lay it on top. So we're gonna use it again right off, right?
Yes,
Yeah.
and we're gonna fly straight up, so
Okay,
it should be
so
fine.
I'll just put it in the box, and then I'm not gonna holster it.
Mm-hmm.
And then I'm gonna grab the page.
Do you need me to come out more on the torch before you do that?
Sure.
I'm pretty far out for, yeah, I'll come out more. But getting close to that. Probably all that should go. What do you think? That's good. Okay. So do you.
Ready?
Hello, are you ready?
Actually, I'll have to holster.
So one time wasn't Pete talking about like binding together the temp probe and the pH logger?
Yeah. Or the super wand and the pH logger.
That was it. That was it. Yeah, I was gonna say because they have similar temperature tolerances.
Yeah.
It's kind of non-trivial, but it kind of might save time. I might mention it to him again. I think you'd have to like bore an angled hole through the, maybe make a new cowling for the pH logger and like get the nozzle in from the side.
Yeah, it would be nice.
So we rush back.
Alright, super flow completed. Now I'm going to pump RNA later. Yes.
Yeah, I'm a big fan of what a scientist I used to dive with a lot. My PhD advisor would call studio lighting, which is where we would do one-sided lighting, usually with the uppers, sometimes with the mids, depending on where the subject was. Here we don't have independent side control. I appreciate feedback about lighting. It's a new vehicle, and the scene setup is going to be different.
Okay.
Unfortunately, I also don't have very many manual control, camera controls in my fingertips directly. So right now we're deploying an optical pH sensor to get pH data from this vent fluid alongside the temperature data and the genetic data that we'll eventually get when we process that filter.
No, in situ RNA. Is RNA later.
Oh, I thought it was is RNA later as opposed to isn't RNA later.
Well,
Like not.
it's double.
Yeah. Ah. You usually nick them?
Yep.
Have a break? I don't know that. What kind of pHs have we been getting from near orifice, Rachel?
Like 6.3 for
Cool.
for like our samples. Jill, I think, has seen like down to 4.4.
From the RTT fluids.
Yeah, the end member fluid.
Yeah.
Just close to the kid as well.
Yeah. So the fluids coming out of these vents are pretty strongly acidic for anything in the ocean.
Is this angle gonna be alright? Is that too perfect?
Rachel?
This is fine. I mean, just do the best you can.
So if you can hear Rachel from the logger mic, she was saying that the end member fluids that we get out of these chimneys, which is to say kind of the most crustal fluid-like fluid before it mixes with the seawater, Jill, one of our co-chief scientists and geochemist, has measured pH of the recovered fluids down into the 4s, about 4.3. That's pretty acidic. With these in situ pH measurements that we've made near the orifice where it's mixing with seawater, we've measured pHs down to about 6.3.
That's probably okay.
So look
You
when
have to keep it there. I don't want it to get much hotter than that.
This pH logger isn't interfaced directly with the vehicle. You'll see there's no cord. It's just logging to an internal memory card.
We're gonna sit here for
So unfortunately, we
5
can't give you
minutes.
live pH.
Does it need to be in the flow?
We don't want to cook
we
it.
don't want it to get too hot.
Shouldn't be too hot, it won't pretty hot.
Yeah, we don't want it to get above 40. So this is kind of like right at its tolerance. If you want to try to get it a little bit closer, you can.
We're a couple inches out of the flow right now.
Yeah.
And I know it changes, but
Yeah.
we're
No.
off to the right.
Yeah. If you want to try to get it a little bit closer, just try not to get it below the super wand.
How's that?
That looks good in the stick.
That was good.
Rachel, the objective list doesn't say how far up we fly for the mid-buoyant plume, and I kind of forget.
I'm wanting to fly up to temperature range of between 6 and 10 Celsius. So
Okay.
It's not gonna be that high.
there's a bit of a disconnect between I
Yeah.
think like what I want as mid-buoyant plume versus the rest of the super team.
Well, you're here in the control room.
But
Last time we
these
flew
are my samples.
Yes.
So
Last time we flew up like 20 meters, and I
Yeah,
was sort
that-
of powerless to stop anything.
yeah,
Um,
it's fine.
but great.
Just on 20 meters or not.
But 20 meters, there's no anomaly. Totally undetectable.
Yeah, I
I
think
think-
the rest of the super team is thinking about distances in meters, and I'm thinking about more temperature gradients.
So you said how many degrees C?
For the next one, I want between 6 and 10.
Okay, so if we could reach that here, like a meter above it, if it was 6 and 10, that'd
That's
be fine.
great. Yeah.
So we'll get a feel for how quickly it falls off. So it's a pretty small plume. So
Yeah.
just
Oh, the difference I didn't really explain between the end member fluid and the fluid that's mixing is that the stuff that Jill has been sampling with the IGTs, she gets by actually sticking the straw some distance into the chimney, which is something we would dare not do with these in situ sensors. It would just cook them. It wouldn't fit. So that's essentially the difference. You know, it's when we sample from down in the chimney, it really has barely mixed with seawater at all, aside from what's pulled in through the rock by the Venturi effect.
One more minute. Okay, we can pull the pH probe and switch out to the temperature wand and then move up.
Yep. Not above 40 C.
Is that Pete's handwriting?
Yes.
I really like the branding of your tape. I'm like, okay.
I think that's a sticker.
Yeah,
but
it's
if we were gonna get lab swag, it should be like white Super 33, the logo on it.
Oh, that is very on brand. Custom electrical tape. That'd be a great gift for Pete's birthday.
I wonder how much it would cost. 3M can charge whatever they want. They have the patent. I kind of want to roll with assorted Pete-isms.
Oh, that would definitely cost extra. What would the Pete-isms be? If I
I'm not sure
put
all-
my friend's hat on?
I'm not sure all of them are broadcastable. They're all well meant, but I'm not.
If I could wave a magic wand, that's another one. That's another Pete-ism. Most parsimonious explanation.
So it was 6 to 10 degrees is what the next sample.
Yes,
Needs to
so
be.
just move straight up in the plume.
So we're gonna come up and then just get a feel for, try to stay in it, get a feel for what the temperature gradient looks like.
I think so, but I also think it have to do with the
On
dissolved metals
sensor, you should be all right.
present in the fluid in this particular area. That's something Rachel might be able to speak to a little bit better. Yeah, we saw a nice Phaedarian, aka Radiolarian, for a long time yesterday on the way up. 2 days ago, Tuscaretta something.
You might need to come towards us a little bit.
We're going to study up on this temperature point, and then I'll ask Rachel about determinants of smoke.
But it's still going.
This is pretty good. I'm happy with this temperature.
I think you're a little. Well, it just depends which way it's blowing.
Yeah, it might move around and you'll end up with something lower.
Or higher,
That's-
because it's getting blown into it now. We can hop up too if we can reach.
How's that? I thought the same. Going into the stream.
Yeah, well,
This seems reasonable.
Happy with this, Rachel?
Yeah, I'm happy with this.
alright, so I'll match with the other end.
Frozen?
Just want to watch the temperature one for a bit. It's probably okay.
Touching. It's perfect. It's touching. Inside the water.
Okay. I'm going to start purging. It's 2. It's 1. Alright. Pumping for 20 seconds.
Good morning and welcome back. Yes, we're on the same vent from yesterday. We call it active site 3. Today we're getting genetic samples from a couple of different points in the vent plume that we got chemical data on yesterday.
Mid-buoyant. In situ.
We're pretty sure Science Crab is still around, but is feeling a little bit less social today.
Come on.
So Rachel, is it predominantly like the crustal fluid composition or the amount of mixing that goes on by the Venturi effect, or something else that determines whether a vent smokes?
It has to do with the temperature of the fluid and the dissolved elements, like trace elements that are in there. So the reason I think this is, Jill would be more correct in this, but the reason why I think like the little chimlet was very hydrogen rich is because it was a very young, small chimney. So hydrogen is very slippery and is one of the gases that diffuse away the fastest. And so you're not going to find hydrogen in a big billowing black chimney because it's long since diffused out at the bottom. Chimneys are super porous. And so the black is a bunch of like reduced iron that is being it's dissolved out essentially from the fluid itself, and so it's coming out.
Thanks. Helps clear up some things I was wondering about too.
Okay, purge complete. Now sample for 700 seconds. Starting now.
The event Rachel was talking about was the chimlet from Pinguinos, which, to my minimally trained eyes, looked similar. We probably have hydrogen data for this event because Jill ran the fluids last night, but I don't know that any of us have seen those data.
I haven't seen.
I haven't. I'll know soon enough.
I do know that this fluid is actually very salty. It's briny. It's like 44 PSU. Seawater is like 34. So this is, Jill said this was the briniest seawater that she's ever sampled.
Interesting. But I imagine those salts are soluble enough that we don't see them crashing out as smoke. Well, I mean, that's sodium chloride, which is highly soluble.
No, but it probably means that one of the reasons why it's salty is because of radiolysis. It's literally like radioactive decay splitting water molecules, and so you're losing your water in from the system, and so it's becoming more saline.
Oh, wow. Interesting.
So where does the hydrogen and oxygen get dissolved into the water?
Yep, and it just dissolves out really quickly.
Wow.
As molecular hydrogen and oxygen.
Yep.
Huh. Very cool.
Sea log timestamp on this.
So that one was 11/27/24.
Okay,
You ready for the lat long?
ready for the lat long.
Okay. 2.099 421.
Okay,
Longitude -91.
yep.
906 109.
No. It was it's such a good
Are you keeping track of temp anomaly for this? I don't know whether you
Yes.
are wanting to. I guess we'll wait till we're done and then we'll find out what it is. I just figured I'd write it on the sample sheet.
What time did pumping begin for the sample?
Pumping began at 11:29:52.
Okay.
There's some muscles in that crack. The limpet too on the right. The kind of thing that worm's a nice shot. I should look up a bit about these amphipods. I wonder if they graze. They probably graze on mats.
It's cruising. It's cool.
Yeah.
It's amazing how mobile limpets are.
Yeah. Yeah, when they want to move, they move, and when they don't want to move,
Get drawn in.
I actually wonder if the limpets down here have home scars. So limpets in tide pools have these really interesting behaviors where they're territorial,
Right.
and they'll sort of farm a patch of algae, and they rotate around it, they'll graze it in roughly sectors, and then they have a particular spot on the rock where their shell is perfectly formed to.
So
Like that's their home scar.
Grow into it.
Exactly.
Oh, that's cool.
And they can home to it with remarkable accuracy and repeatability. You can pick them up and move them, and they'll find their way back to their home scar. Some species, owl limpets.
That seems.
That experiment's been done with.
That's incredible.
And I wonder whether there are analogous behaviors for grazing on bacterial mats here. Somebody in our lab, Ian, maybe you met. Yeah, he's kind of interested in how these make their living. He thinks that
in terms of like some sort of symbiotic
symbionts potentially on the shell or elsewhere.
That's why he wanted us to vacuum some like this.
Mm-hmm. We have a bunch. There's like 70 of them from one of the live sulfides yesterday that are waiting in the cold room for me to freeze them individually.
We didn't put any live sulfides in mason jars, did we?
I put a hunk in one mason jar. It's very full. The other one, but I haven't frozen the other one. It's just sitting in anoxic seawater.
Okay.
Go back in there a little bit.
Yeah. Sorry, our worm guy wasn't watching. Our worm go backwards.
I wasn't. Too shimmery.
Sort of. Trying to tell.
Take it away.
Oh no. Well, maybe I will, but I like this frame well enough. I'm just trying to tell what that is up there.
Unfortunately, we're at full zoom.
Yeah.
That's
See,
why.
so that would be futile anyways.
I can increase lighting and stuff.
That's.
I kind of like the shadows.
Yeah, so do I. No, I'd much rather not flatten it. I'm a big, well, I think it's more necessary in the midwater, down here. It's just kind of a matter of aesthetic, but
Independent.
I love
size.
independent, yeah.
No, it's,
left-right
there's
control.
definitely, if you could, being able to do light groups where you can create your own, like individually plus light groups, like defaults for different angles is nice.
My recollection is that some of the DSPL offerings, the controller, basically the dimmer, is in the light unit.
And you use,
So they're independently
485
addressable.
to-
Yeah.
Yep. And there's also different ways of them, like even the ones that aren't, or sorry, you can still vary voltage and things like that. And
Do you have dimmers on any of these lights?
just the cabinets.
Got it.
I mean, the DSPLs, I think they're dimmable,
But-
not would.
Ooh, what's going on back here?
How bad?
It has a big orange spot. I mean, I think we'd have to pick up to see it, or we just kind of have to wait. It went behind that.
Taking the focus back there.
Well, it's hiding behind this chimney now.
Okay.
So, unless it washes back the other way.
Focus right back there now.
Yeah, let's just stand by and see what happens.
You want to switch out pH to pH probe?
Are we done pumping? Or are you
Not
going to do it
done
mid?
pumping,
Okay, you're going to do
but
it mid
just
pump.
Keep pumping and switch temperature 1 pH probe.
Ben would love to.
Thank you.
Oh, there's our worm friend. Come wide though.
Are we gonna use the temperature probe again, Rachel?
Yes.
So I'm just gonna put that.
Do you want to do it near chimney next?
Yes, that's what I was
Okay.
thinking. As long as we have the super on, we'll just take those samples. And then we'll come back and take a sulfide.
Are we basically reproducing yesterday's samples with RNA later, with the exception of this one, the midpoint one? Like, do you want to go to the same place for the near chimney? Is what I'm asking, because we have a marker on it.
Yes, do that for near chimney. Yes.
At what altitude?
For yesterday's,
Well,
it's the same altitude as the vent orifice. So
Okay.
1669, I believe.
okay, 16. Yeah, depth 1669. Thanks.
Yeah, I think we said it was. yesterday.
Yeah, it's right here. I think the tracking's off a bit from yesterday, which is not surprising. So, you know, if we end up 4 or 5 meters to the southwest of that marker, it should be the same spot.
I'd only be worried that that handle's going to rotate down into the hole. But I think you just come right back and let the T handle, let it rest on the T handle. Just make sure that right a little bit. I don't know. We'll see. That might be optimistic.
There you go. Grab that again.
Oh, well, that was close. Good job.
Yeah, Nicole is working on a genetic barcode library for vent-associated, sort of vent-proximate animals. That's why we've been slurping a lot of the animals, mostly from diffuse. We've been slurping the animals from diffuse seeps, but also from the sides of, or diffuse flows, also from the sides of some chimneys. And she's also been collecting water with all the Niskins. That's why we've been firing all 6 Niskins at once. She just gravity filters that water out of the bottles and sequences it. Of course, if she doesn't have reference sequences from animals that she's visually identified, you can't tell what's in it. So building and testing the database is one of her main objectives.
Okay, super complete. First one is switch over to SV42. I'm gonna pump a little bit shorter this time, do 13 seconds. Okay.
No, it's no. Yeah.
Okay.
We're really focusing on vent-associated communities on this cruise, so I don't expect to be taking any collections or even DNA samples way out, you know, even outside of any individual rift. But a lot of you on here have probably seen that we have been sampling diffuse flow mussel beds that aren't immediately next to active chimneys that are just part of the vent field. Of course, because we're interested in things that are specifically associated with hydrothermally active areas, we do have to take background controls. And so these super samplers, which are also genetic samples, the only difference is that they're filtered in situ rather than up on deck. When the ROV comes back, we do take background samples that are at least 10 meters away from the active chimney we're focusing on. Sometimes more.
All right, sit here for 5 minutes.
The DTS transects are another exception. Philip is actually going to be running transects, I think, once on every vent field we do. So those transects are at least 100 meters a leg, and we can reasonably expect to see some colder water life along them. They always center on a vent.
So the temperature wand, that's what it's measured on the temperature wand, and it's back in the basket. So we finished collecting our sample, it's just sitting there, and we're doing the pH probe now. Thanks for checking.
Yeah, it's done.
Pretty toasty down there in the basket.
So you can see the cables like falling over on this side of that holster. I don't know. But when are we gonna use the temp probe next?
We're gonna do our near chimney sample. So yesterday we said we put a marker down for where the near chimney sample was taken, so we'll move there
Yep.
and take a super there.
Okay.
And then we'll come back and grab a live sulfide piece to put in the RNA-later column liver. Does that seem reasonable?
Yeah. I was just wondering if we needed the temp probe before we're flying somewhere.
No, not yet. Yeah.
But yeah, that makes sense. Thank you.
Cool.
Do you know
Rachel,
what the name of the marker is?
I think yeah.
super near chimney.
Super
But
near-
let's head about. I mean, based on what looks to be the nav drift since yesterday, let's aim like 4 or 5 meters southwest of it.
Okay.
Well, we're wanting to be about 10 meters away from the chimney.
Right.
It would still be 10 meters.
Yeah,
So
it'll be
just
10 meters
Okay.
away. It's just that this was the marker we dropped yesterday, and this is where the site actually is based on
Okay.
a visual fix.
Yeah.
Okay.
So we want to, we want to just use the same offset.
Okay.
What would you think, Rachel? Because we're not seeking a temperature in this case, we're just seeking a location. If we did not put the pH probe away, fly over, start the pump with the pH probe, and then switch
Yeah,
to temperature.
that's fine. That's
Okay,
smart.
just save a little bit of time.
No, that's smart. Yeah, we're not, it'll be background,
Yeah.
essentially.
So we are not going to stow the pH probe after this reading. We're not going to stow the pH probe
We'll
just
fly
yet.
with the pH probe.
We have to get the temp.
Are you comfortable flying with it?
Yeah, it's just the
Okay.
temp probe fell, so we're gonna have to recover that.
Oh, okay.
It's
Well,
hanging from the sub.
Okay.
So it's not like, but we'll have to retract the porch and kind of search for it a little bit.
whatever makes you most comfortable.
But, yeah, we can do that whenever.
Ooh.
Use the temperature.
High drama.
So that's why they
You got an eye for this, Alex.
It's like subsea soap operas. It's like the same plot every time, same actors, bad lighting.
Oh, I can comment.
Diplomatic efforts were successful.
It's just that sneak attack.
Not today.
Yeah, I want to know who's hiding in the hole.
Oh, right, yeah. Crab kind of thing out, but past the same. Oh, where is it? Did we lose one?
Lose who?
I see him on the sit. Maybe we can't. Oh, there he is.
Oh,
Oh,
is that science crab?
big
I
crowd.
think so. You can
Science crab?
Signs.
There he is.
In the background there.
Yep.
I do.
just keep and watch.
So something interesting about these crabs, which I think belong to a family called bathygrades.
Yeah.
These are cyanogrades, but that I learned from a hydrothermal researcher in France who has actually kept them in captivity. That looks like a cool amphipod, kind of phrenemid. Is that they need to move back and forth between
It's a little muscle.
a warm spot and a cold spot. He's kept them in captivity. You can actually see them on display at the aquarium in Brest, France, and they have a sort of heating element, which is just a tube with water that's some 40 degrees running through it in the middle of a tank that is otherwise chilled to background temperature, right, 3 or 4 Celsius. And the crabs will move back and forth between the warm tube and the cold water. If you give them an average temperature that's right in between, like 20 degrees Celsius, they won't survive, which I think is really interesting.
Right, we can pick up and fly to the super near chimney marker.
So yeah, 5 meters southwest of the super near chimney marker, just to account for nav offset.
I'm gonna still the arms first.
Okay. Do what you gotta do.
Come back in a little with the porch while you're doing
Good morning, good evening to those who have just joined us. This is SuBastian Dive 0566, our second on the Pinguinos vent field on the Western Galapagos Spreading Center. Right now we're taking some genetic samples and parallel pH and temperature readings on a vent that we took chemical samples from yesterday.
Happy with that, then?
Yeah, but I got some.
We're going to move off the vent site for a little while to take semi-background samples.
I'm gonna use
Then we'll come back, collect some of the actual sulfide, and after that we're up in for some lander ops.
Or are you gonna go all the way back?
Let's put it all the way back.
Okay, come back out. Sorry.
Stay there.
It's right over.
You're here in the Falcor 2 control room with pilots Ben and Alex, loggers Haley and Rachel, and
Yeah.
I'm Jacob in the watch lead seat.
Quite close to the sides.
When do you want to swap, by the way? Next dive? Think about it. Cool.
Yeah, I don't think I'll be doing any supers on the next time.
All right. Well, no pressure if it becomes stressful, but yeah.
No, I'm not stressed about sitting in the hot seat.
Honestly, I think where you are now is the hot seat. Like I said, I'm going to struggle more with that.
Not with the hot mic though.
Mm,
Is that one on?
pretty sure.
Okay.
There you go.
Oh man. Just all these opportunities. Alright, well now that I know the mic's hot. Yes. Hello to my family. I am speaking today.
You're using this right away.
They've been upset. They're like, we can't hear
Get it out of the
you.
nighttime probe.
They're not smell that much. I'm just parroting what people are saying and use.
Oh man, if I started listing those,
Come back in with the porch.
we might be at intermission by the time I was done. Though I'm happy to start, try and explain.
Fish.
Rachel and I, since we're the two who are in here, are
Can we put the,
working together on
You're gonna be pretty close to that.
a project looking at methane oxidizing microbes that actually live in the chimney rock.
Should we be telling them that they
So
are
I'm doing a bunch of biochemical profiles looking from the inside to the outside of the chimney wall, which is a huge temperature gradient. You know, from 100 degrees on the livable inside margin to 2 degrees on the outside. The biochemistry of those microbes is going to be wildly different between the inside and the outside. There is an interesting schmutz ball. I'm not going to catch it.
Or should we bottom left,
Well,
I think?
Tina Force don't even have smiles, so I know.
That is not true. I'll prove to you otherwise.
I didn't see that Tina Force smiley.
well, it wasn't in a very good mood.
Did you exit out of the driver's menu?
So I'll be doing biochemical profiles on the chimney walls, and then Rachel and I have actually ground up some of the chimney walls. We've given them food, which is to say sulfide and isotopically labeled methane, and then we put them into this whole array of culture chambers at different pressures and temperatures, 4 different temperatures. So far we've gotten 2 done, and 2 different pressures, surface pressure and the pressure at these vents, which is about 170 atmospheres, to see whether the pressure has a specific effect on the rate at which they take up methane and on their biochemistry. Let's see, what other experiments? Jill's doing really comprehensive geochemical profiling of the vent fluids. That's what all the IGTs are for. And that's why we have all this fresh hydrogen data and so on for you.
The tip of the probe, I think, is facing the wall there right now, so you might be scratching. Might need to come to
Nicole's building the eDNA library.
What's that? I'm trying to-
Philip is running these community transects off of vents.
or the direction that the probe is going.
Johan has
There you go. Now
has the distributed temperature sensor that was deployed 2 days ago.
Nice.
Amy Gartman and her whole team are doing mineralogical characterizations of all the sulfides, active and inactive, that we break off. They also want to analyze some cores, but we haven't found very many coreable sediment patches yet. I'm sure I'm missing plenty of experiments, and I'm just going to have to get to them later. Hopefully you can get some first-hand explanations from the people doing those other experiments. I can't tell if that's a dandelion or a Schmutz ball in the down cam.
I believe that's a sea booger.
Yep.
Okay, so we're gonna use the temperature probe as soon as we get to that one, right?
That's fine.
I'm just gonna hold on.
Yeah, the order doesn't matter. I concur about the sea booger.
The probes.
Are you adding that to sea log?
Happy for me to bring the portion. You want to set that down?
The Ambari annotation system has floaters and sinkers.
Yeah.
We label a lot more sinkers than floaters.
I'll bring it more and we'll keep the stubborn random.
So we can read the,
I feel like our
suction sound.
our dive, like our ROV watch should have like a morning radio show jingle.
Okay.
Like,
Bring it in.
Yeah.
Well, you're going to be host tomorrow. You better start
I
proposing.
I've gotta come up with a catchphrase.
Yeah.
My mom's in radio, so like.
She is.
Yeah.
We're just going to head up just to the
Yeah.
southwest of
Gotta come up with a jingle.
So Rachel is going to take over as watch lead tomorrow, and we just found out that she's a pedigreed radio host. So you're in for a treat. Also a highly knowledgeable biogeochemist, which isn't a combination you find every day.
Very not knowledgeable invertebrate biologist.
That's why we specialize.
You know, I did take that class in college. That was fun.
Yeah.
Dissected a lot of polychaetes.
That's good. Yeah, I need to.
We looked at the lower part of this yesterday. It's sort of flange-like, but it's diffuse, and it's like all worms, practically made of alvanellids. Yeah. We asked about sampling it, and Amy just said it would crumble.
So we're going
Worms
to
go
head
everywhere.
to the northwest, and we're going to end up roughly 4 or 5 meters offset from super near chimney. Yep.
Bye, Active 3. We'll be back soon. Coming for you.
Oh, the other major goal of this expedition that, I don't know, fall into the category of experiments, but it sure is exciting, is that after we leave Pinguinos, I think we make a stop at Navidad.
Navidad.
Right afterwards, which is the third known event field that is on
Known but not imaged.
our cruise plan. Right. As Rachel says, no one's actually seen it. So we'll have the first ever images of that, and then after that, we'll look for unknown vent fields by trying to trace chemical plumes effectively. I think we're using the oxidation reduction
Yeah.
sensor on the Toyo. Is that right?
Mappers, yeah,
I think,
the ORP
yeah.
with other
The map
mappers
is
and
full.
APR.
And temp anomalies.
Yeah.
Okay. So a whole suite of instrumentation I actually don't understand very well, but you have two experts in the same room right now if you're curious.
I am on the Toyo watch though.
Maybe at least you know what the acronyms stand for. So a Toyo, as a high-level overview, is an operation that involves yo-yoing a CTD rosette. So that's a water sampling platform that also has a conductivity, temperature, depth sensor package on it, along with any other sensors we care to attach. One of them is an oxidation reduction probe. Sort of normal CTD operation. Okay, that looks like station.
So what altitude or?
About the same depth as before, right? One.
We want to go up 3 meters.
Okay. 1673. Depth 1673 meters, please.
1673. So that's 10 meters deeper than we are now, but
Wait. Oh, I'm reading the wrong depth.
Well, we
No,
came-
no, that's right.
we went downhill as we came.
Oh.
Yeah, 'cause like 1669, 1670.
Yep. Yeah, so a tow-yo is sort of like an ordinary CTD yo-yo operation, except that the ship will be underway. So we get kind of a two-dimensional swath of water, an oblique diagonal transect of water with physicochemical data along the way. And Jill McDermott, one of the co-chief scientists, kind of has a track record of using this method to find previously unknown vents.
This is good.
You're right there on the Alex.
Oh, wait.
Yeah, he drove his
More super sampling.
And once we get up another meter, just take the arms off, follow over, let's see if we have the temp probe right next to it.
A lot of those are amphipods.
Are they?
They're swimming. Well, the ones you can see them in the
Yeah, I
sit
can
cam
see
better.
the- yeah.
Oh, yeah, look at that.
I think they're amphipods. They could be some other crustacean.
You said amphipods.
That's as good a guess as any.
Not marine snow. Settling particles from the chimney.
Considering we're looking down, that's a possibility. The trails kind of have me thrown off. Yeah, it's hard to say.
For the purposes of my publication, I want this to be a settling particle.
You want this to be what?
A settling particle.
Okay.
So we've determined.
We have determined
This
it
is a
is
settling-
on the record.
Yes. My official scientific opinion. 2 out of 3 here. 2 out of 3 scientists agree. Not animal.
Not animal poo's fine.
Yeah.
So whenever you're ready, I'm holding that 1673.
Are we ready to sample?
Yes.
All right.
So all we need to deploy the ion.
Alright. Bypass pump. Starting now.
Strangely enough, even though we're very near where hydrothermal vents were first found, kind of between the East Pacific Rise and the Galapagos Rift,
Where's this super
Where's the super nozzle?
We're getting
Okay.
whatever.
I learned earlier on this cruise that apparently no geochemistry data has been taken on any of these vent fields. So those data that Jill is getting from the gas tight samplers are the first we have from this whole area. And it's nice that if we do find any new vents, we'll be able to characterize them pretty comprehensively.
Here we go.
Thanks to all of Jill's kit.
There she is. Super.
Move this down.
Clutch.
Beautiful.
Nope.
Is this,
Yes. Yeah, well, this was just very strange. Okay, purge complete. Oh. 700 seconds pumping now. What was the pump start?
12:06:23. The actual time is 12:04. You want your coordinates?
Yes.
Okay. Or 2.09.
Yep.
9541.
Okay.
And then it's -91.
Yep.
0.906.
Yep.
2.
23. Okay.
Yep, that's 906.
May I?
Yep.
Okey-dokey.
You got it.
I doubt I'm gonna-
Right now,
Yes.
anyways.
Cool. I'm not gonna see anything, but
Zero.
yeah, both of them surprised. Set that on top of this.
Go manual focus if you're gonna be.
Yeah.
So you can go to bed after this.
Oop. Okay, we're manual.
Do what? Oh, land.
Stupid
Oh,
land.
stupid land.
Interfering with my sleep schedule.
No, I think the ocean is interfering with your schedule.
It's fun, man. I'm so happy ocean. I can't believe you're in Stockholm so bad. Like, hold on.
Hmm.
Is this like one of those situations where
This is like midwater collecting.
water until
Like, oh yeah, that little white line.
Sand.
it's gonna be one of those situations where we don't ever
Probably. Yeah. Since we launched it, yeah.
so yeah.
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 Yeah, we should have a pool for when they recover.
I feel like Jill just needs to sleep for 24 straight hours.
That's like an underestimate
Yeah.
by the numbers.
I don't know how she's functional.
We are going to have to switch. We're going to have to switch instruments. So I'm doubtful, to be honest. But let's see if we can give it a look.
What's the sea log timestamp for the start purge?
Ooh, that is interesting. Those are bubbles. Those are oil bubbles.
Oil bubbles?
It looks like you have a no.
They're tears. They're manipulator tears.
Tears.
Ah,
That's
Very cool.
not ideal.
That's fun.
is that oil from Sebastian, or is that oil from seafloor?
No, that's
Hydraulic fluid.
that's manipulator tears. Yes.
You're doing great, Sebastian.
How much longer are we doing this sample?
Yeah. How's our pumping progress?
We've got like 9 minutes, guys. You gotta
Calm down.
I know. I know there's not much to look at, but
Sir, comps, what?
you can watch like little particles like fly into the super wand. It's fun.
Wonder if that's a piece of a worm mouth part sticking around the super wand, or a ctenophore tentacle. No.
That might be your ctenophore.
No, it's different. If it's from a ctenophore, it's a different ctenophore.
Oh, okay.
The expert is
Thinking
spoken.
though.
He's like, "That's not
Well,
what
that ctenophore had orange tentacles. Spent a lot of time freezing in the cold room looking at those tentacles.
Just got the song Bye Bye Little Sebastian in my head. Parts of that.
Yeah.
Bye Bye Little Sebastian.
You're gonna be a great
You should play it next launch.
That's gonna be our jingle.
There you go.
Capped at 15 seconds though.
Yeah.
Yeah, there you go.
It won't take much.
Sorry. You want to split up the comps, please?
Like 5,000 candles and it's the Donner Party with Jacob, Haley, Rachel, and Will Sebastian.
A little perk on
Wilbur.
on Nautilus, like they literally have a little perk.
Oh.
We need a little smash.
Yeah, we do. Need
Yeah, I think they've been.
I want a Will
Well,
Sebastian
y'all have been
sticker.
talking about getting a second ROV, no? I feel like I've heard word about that. Okay,
Rumors.
big
Not
shrug.
confirm or deny.
Rumors. All right.
If SOI were to invest in a second vehicle, do you think it would be a full work class or a smaller supporting vehicle?
I don't know.
Yeah, fair enough.
Will Sebastian.
It's gotta be.
Spread your holy wings.
Wouldn't mind avoiding. No, never mind. That's fine.
Avoiding something?
Look at whatever you want. That's fine.
Oh, what do you? Is this the glare off the?
No, do what you
Oh,
want.
sure.
Don't mind it. Maybe it'll be better. Maybe.
I'm gonna stow temperature wand and pull out
Oh.
huge probe.
So it's in Ecuador, right?
This
Or-
time we stow the wand for good.
You want me to send her up? Would that help?
I'm gonna follow him around.
Great.
Unless
Okay.
you want to.
Well,
Help
it's,
him. Why not?
sure.
You can help him.
All right.
So with other folks on, what you do?
Yep.
We'll just get your. He's gonna be going between where he is now
Uh-huh.
to this white tube. on the
Got it.
front of that
Uh-huh.
suit. So I'm gonna just bring the porch out a little bit. And you're just gonna follow him in
Sounds good.
and get him good situated.
Mm-hmm.
This is great. I'm outsourcing my job.
Yeah.
You're coming out just a little bit to get a little bit of go. Okay. Oops. Now I'm directing that station.
Well, that's not helping.
And if you need to adjust lighting or whatever so you can get some contrast on it.
Yeah, I tried irising down. It's still just washed out. handle overlying the basket?
Not really. We can fall straight down. So it don't matter. If you're wondering why it picked up that way, yeah, that was something magical dropping the probe shed.
Okay. Drop.
I mean, it fell.
I saw the amplifying. Did not see it land. Interesting.
That's not going anywhere. And then I gotta bring the torch. I need to bring on your blowers. Bring the torch all the way out. I could have done that for you, so that's right. I like where you want. I'll take.
You want me to center on the left?
Nope.
No,
So,
okay, hold me.
I'm going to first get this all the way out. I don't know if you reach it right now with the camera. I think I should be able to. Yeah. So I'm going to be stating the pH at number 8 or whatever. So let me get it right out. Maybe a little more set.
Sit cam just stand for situational.
I believe so. I've always just assumed that, never explicitly asked. It's labeled sit.
Mm-hmm.
Get close with your elbow.
Not.
Not enough at all.
Where'd that?
Carboy? Hold the other side? Just to try to catch it.
How about you?
Works a little bit, just
How's it goes it?
Oh, good.
It goes as well since, yes.
What are we playing right now? Midwater.
We're doing jar
Midwater
ribbons.
Midwater
Midwater Super.
Super. That looks like it's gonna be going.
That's how it goes. We're very serious.
Are you doing background? Or?
Yeah.
That or
Yeah.
swivel.
Near chimney, same spot as yesterday, but we're doing in situ preservation this time. Here, before I forget, I brought you all Tinophore merit badges. It's the wrong one, but it's red.
I like that.
Oh.
I have got more of these.
Oh, beautiful.
You've seen, you can have as many of these as you want, Drew.
Yeah, I'm pretty sure they're from your like
Previous.
Yeah.
Yeah.
I think they still have some.
Yeah.
I have more, you know,
Did you
for
ever get the one to glow?
everyone. Oh yeah.
Yeah.
I have some video. The glowing video is so-so. My camera was being kind of recalcitrant, but it glowed for everyone.
Tyler said he got a good video of it glowing.
Oh, did he?
Yeah.
He said he didn't get a good one when he was there. He said he took the camera down right when I poured it from the sampler into the tank. But I'm sure he got some. It's an interesting species. It and its relatives, they glow in the body, and then they shed this luminous mucus. It just looks like a constellation of glowing particles.
That's awesome.
Yeah, so like the whole bucket was full of that stuff by the time I moved it out of the bucket. It was awesome. It also turned out to be actually pretty light resistant, which is something I've observed in one of its relatives also. Most ctenophores shut off, but it varies by species. And so next time I probably don't have to do the whole garbage bag in the dark for 2 hours charade if we get another one. But it's pretty awesome.
Okay, pumping complete. All right. We
Did you see
can-
what we did to your wanted poster?
I did.
We can
I did.
stow,
There's a whole posse of them though.
we can stow super
They're still out there.
And pH probe, and go back to the active 3 to sample a sulfide and RNA.
No, I'll have fun.
Okay.
That works pretty stable.
You'll probably have to repeat that because of the turnover.
The tariffs dropped out. Yeah, well, it's been doing that for a while now.
Okay.
Just normally it's been over like 2,000, but I guess it's been down here for long enough.
Okay. Is the super pumping all done?
Super pumping is complete, but don't turn off the power yet.
Okay, keep the super power on.
So we could stow the pH.
You can stow the wand
And the pH? Or
and the pH probe.
It was not a benthic ctenophore. It was a Mertensiid sidipid ctenophore that we collected on a scent yesterday. J-Rod did a really masterful job of slurping it. Most of the benthic ctenophores, if not all, to my knowledge, are non-luminous, which is an exception among ctenophores. Did you hear Rachel's instructions earlier? I think you were turning over time. So we're, yeah, now that that probe is secure.
You can turn off super power. Super power.
Down.
Super secure.
Okay.
Yeah.
Super duper.
So now we're going to go back to what would be active 3, but it seems like we have a few meters nav drift since yesterday. You can see the cluster in the snow trail here. It's to your southeast.
Mm-hmm.
And we want to head for that cluster.
Okay.
And set it back on the chimney. We're going to sample it.
Roger that.
Oh yeah, she never. I don't think she. She's not been sleeping well.
Do you want me to porch in a little bit, or you go out
And then I wake
there?
up at 3, so she might wake up at the same time I fix up.
It's totally up to you. I'll just gonna get you set up and then I'll
This morning
start
she's
driving.
So
So pretty much what I've been doing is getting the green like roughly in the middle and black
printed it.
in the channel, and then slowly opening. You got it all in one piece.
Not helpful.
So that's it. I've had issues with it not wanting to go in. I had to roll with it.
You have to be normal.
Thank you for logging while I was
Supering, yes, no problem, anytime.
Okay. Start porching back in. And what are we doing over there?
We're going to sample sulfide
Naturally.
into a coral quiver.
into a coral quiver.
I'm sorry, where were you?
That little,
Yeah.
my little.
They saw some benthic ctenophores on 2 cruises ago on the EPR. I would love to find a few of those here.
Here. Not this guy.
Rachel, is that our target area? Oh, or a little.
I don't think this is the right.
Akimbo here.
That's not where my cluster's at.
It's true.
Keep
maybe it is.
rotating.
Come around it, yeah.
It might be, yeah.
That's a very obvious spire.
Yep. I'm getting closer to it.
Pretty sure it's that one, right?
Oh, yes, yep.
Right
yep,
down here,
there's-
lower right.
Yep, there's its little
See the little smoky bit?
Vegetoa toupee.
So that, the skinny one, right?
Well, that's yes. The active chimney is right there.
Yeah,
That's where we want to grab.
The basin.
He should be.
Okay, cool. So I'll print some more out.
Right, to the heart.
I wonder if the crabs have polymorphic coloring, or what, or if they're different species.
Well, Nicole was saying that the young crabs are more orange.
I could see that, but I've also seen some very small crabs that are white. I mean, there are all kinds of different explanations. They could be sexually dimorphic. Yeah, you know, females are smaller.
Their diet.
Yeah. In short crab, there's a lot of just color polymorphism. Like that one at lower left that we just passed over with the arm, it has like a white patch right behind its face.
There's science crab.
I wonder if I can rest on that stuff.
An interesting couple of days for science crab. Biology observation, science
I think science crab would deserve a marker if this area wasn't already so so saturated. The toupee is interesting.
It really
Do you think there's any chance there's a leak there? Or you think it's just eating the, like, just...
There could be a diffuse flow. It's hard to tell.
Yeah.
Do you want it?
No. Well, kind of.
We will.
We might as well grab that inactive before we leave.
Do we want to put it in RNA later?
You can put some in RNA later if you want. I'd be interested in having it in the bio box. The piece I got yesterday was not very big.
Okay.
I didn't know we were coming back.
No, yeah, but for sequencing purposes,
But
I don't want a big giant
of course.
fluff ball. That's just
Yeah.
gonna
Is this
swamp
the
my
thing we want?
reeds.
That is not actually the thing we want. The thing we want, primary objective, is that stump,
Yeah.
the active stump.
Okay, right here, we're low.
Yep. And that's going into a coral quiver.
Do you want this tall thing?
We do.
Do you want to get that first, just in case when we start messing around the base of this, we don't accidentally knock this whole thing over?
Is that all right with you?
Yeah,
It's not the primary objective, but it should be quick. Yeah. So we're gonna try to get the tall inactive into a bio box, as much of it as you can.
As much as we can.
Yeah. And then, do you think it would be? Do you want some in RNA later?
Yeah, that'd be nice.
Okay. Do you think that we should go at it initially
I'd say
somewhere like where?
Well, you can take off your piece from the top,
All right.
put it in the bio box, and then the next piece down can go into a coral quiver.
So where do you think we should make the first grab?
At the top.
All right. Yeah, that's a big enough chunk for me. Like maybe below that Veggietoma mat, right?
You want?
No, below the lower one.
Oh, okay.
Does that work for you?
Yeah, perfect.
There's
always a chance that I could crush it.
The whole thing.
Or drop it. So
Yeah.
if we take smaller sections, we have chances of going back for more.
Okay.
If we,
If I fumble it.
that's fine. I mean, I think there's plenty to go around here.
Okay.
That seems like a nice increment to start with.
I feel like you're gonna go grab it and this whole thing is.
It's just gonna crack over.
Yeah, no, that's entirely possible too.
Because you want a little bit. If you wanted the whole thing, you just all shit.
Well, yeah. Maybe you just aim for that mat and grab it right there.
Okay.
Target.
Rub it on? You can use port. I was just wondering which arm so I can get
This one is port, but
That's the way it's leaving.
well, so
I mean, we're going into the bio box with a section of it, and we're going into an RNA later coral quiver
With a smaller section of it.
for the smaller section, which are both on the basket. Those are the only two places we're going with it for now.
Correct.
Mm-hmm.
Yeah, I think it's started on. Does it matter which bio box it goes in?
No, they're both empty.
So I'll do starboard on, and probably shoot for the middle bio box.
The middle bio box.
Well, the one in the middle of the basket.
Hmm.
Graphite. There's graphite.
Oh yeah. That's Bedutoa. Yeah. Which we find curious because I think it wants vent fluid, and this inactive chimney doesn't appear to have any.
It might be a very diffuse flow.
Looks like
We'll see when we crack it.
pretty close to the thickness of your arms right there. So it's a camera.
Sample ID, this is rock 1. Chimney site, active 3.
Nice. Uh-oh.
What?
Might want to go higher.
It just started to kind of timber over
Oh.
a leaning tower. It was just a little bit wide for
I
the
would
jaws.
try just above that mat,
Yeah.
just so you know you can get your jaws around it without tipping it over.
Oh. Okay.
Oh, that's good.
Alright.
We can work with that.
You don't want the port arm for this now?
What do you? Oh no, you want something from the lower part,
I would
Rachel.
like the lower piece.
So why don't we take the top bit above the crack and try to go to the bio box with that?
And, you want to get the port arm up there?
Sure.
It's like a wizard staff.
I think I
I'll try to
see
get that top
some.
part.
Okay.
It's hard to tell with all that shimmering down at the bottom, but looks like it might be leaking a little bit. See it?
Yes, that's definitely diffuse flow.
You want me to try to hold the base and stabilize it?
I don't think so. Can you make bits in there?
That may have already sectioned off.
Yeah, it looks like it.
It's going to bio box 1.
I'm not sure yet.
Yes. Well, that was
The plan.
the idea, but
Looks like those jaws have like almost a centimeter of deadband. Kind of jerky.
Nice.
Don't.
Yeah, we see a lot of fallen chimneys around these vent fields. What is that?
There you go. There you go.
No, it's still not there yet.
Oh.
Looks like a borax. Estimated size.
2 fists.
Follow me with the fork cake, you know what?
efficient spacing.
You want another piece of that?
Yes, but in the coral quiver with RNA later.
Do you have a timestamp on our 01?
Yes. It is
What's that?
12/31/04.
Thank you.
Yeah.
104. Oh, we should. Did we get a good?
Yep.
We should get a good frame grab of this one too.
We got both.
Great.
Okay. Do this one too.
I'll try not to zoom in too much so you can have a view.
I'm assuming the one furthest away is going to be the most immediate.
Yeah. Do you need the forte or can I take it?
Screen grab. Thank you. Yeah.
Cool.
Look at that RNA later.
Yeah,
It is
still there.
still there.
So this RNA preservative that we use is basically a saturated salt solution. It's mostly made of ammonium sulfate. And it's really good at inhibiting the enzymes that break down RNA, which is an information encoding molecule that's really unstable. There's also some EDTA and a citrate buffer system in there for all you chemistry geeks who are watching.
It's all going to get it.
Don't see that being this area.
Oop.
Oh.
Oh, but that's got veggie toe on it. You okay with that?
We'll do what we can. Take what we can get. That's still, yeah, that's fine. Sample, rock 2.
Could just like clip sections of it.
That's exactly.
Yeah, I was tempted to get greedy, keep piling it into the bin, but it's going to take a while. I don't know though.
I feel like this is going at a pretty good pace.
If the Veggietto is
Oh, the veggie toe. Well,
problematic, I mean,
I defer to Rachel on that. If you wanted, we could toss this one in the bio bin. We can go back for another piece for you.
I would get one in each.
I can
first.
always subsample this.
All right. Nice size chunk.
That's pretty.
That handle's in the way.
alright.
Okay, that's okay.
So fluffy.
A little bit more than that, please.
Fluff fest. Those of you who don't know, Fluff Fest is in Somerville, Massachusetts,
What handle?
and it is
Oh,
where
that's the rocks.
the marshmallow fluff
Mmm.
was invented.
Somerville?
In Somerville, in Union Square. And so they have Fluff Fest every year.
Seen some ads for it.
What is it?
Personally, I think it's a strange thing to celebrate, but the invention of marshmallow fluff.
No.
There's weird things.
That. Look at that chalcopyrite.
That's a temperature gradient right there.
It was a temperature gradient. Do you think it still is?
No.
Nice.
Oh, are we gonna have to kind of? What do you want to do about that, Rachel?
Oh yeah, y'all got that drop shot in there, huh?
I don't think. Yeah.
You just like push it down a little.
You want to
bit.
just push on the lid and
Just
crunch the
tamp-
end a little?
Yep.
All right.
Alright, that's all you if you don't mind.
What core quiver's that? 5?
That's the one we're talking about. The one in Somerville, Massachusetts.
Alright, so you just want me to
Have you ever had a fluffer nutter?
try to push
I
it
been
of
a
course
very long
had a
time.
fluffer nutter.
When I-
Do I even live in Boston?
One of my friends when I was in the Navy was from Maine, and she was always talking about fluffernutters and whoopie pies.
Mm.
Y'all name things very strange, but they do taste delicious.
They are.
We're at that time of the cruise.
When what?
Start talking about food
Food?
excessively.
I think the stuff in the mess is great.
It happens.
Yeah.
It's funny.
You're gonna miss pizza.
Yeah.
You can't make pizza on the ship?
No, they don't have enough ovens,
Oh.
and so whenever they do make pizza, they like to make it towards fresh and coming out a lot.
Uh-huh.
And they only have one fully operational pizza that can get that hot, or oven.
Well, you
that
have
can
the
get that hot.
grills. You could like do flatbreads on the.
They'll make flatbread
Yeah.
stuff, yeah.
Yeah.
But, like they can actually make really good pizzas on here when they have both ovens going.
Oh, I'm from Waltham. What's up? unless that's Matt.
Do you play D&D?
I have played D&D before.
Is this a friend of yours?
I don't recognize this person, but yeah, I live in Waltham.
Personally, my favorite products of Camberville are Junior Mints, and
Oh, I love
Deposit
Junior
chocolate.
Mints.
uh,
I like
to be
the
clear, we're not
popcorn.
advertising or endorsing any particular products here.
A disclaimer, did you hum?
We're just hungry.
I'm from
We're just
Piety
Not
Corner.
product placement.
It is 18 minutes till
We're just
breakfast.
hungry.
We're just,
All right,
Just really
just
hungry.
just to be clear, so I'm just gonna take the lid and kind of
yeah, just
grab it down.
yeah.
All right.
The other piece of chimney that we're supposed to be getting after this,
The active one?
Yeah, that's going in RNA later?
Mm-hmm.
Okay.
Are you?
And nothing else is going in BioBox 2?
No.
For right now?
No, I don't think it's necessary. Are you gonna grab the eye or what?
Just grab the handle and set it down. And you can push down on
Press
top.
on it.
Yeah.
Just
If you-
tamper down. Now, are your D&D campaigns about science? My goodness.
You might have to push on the metal a little bit. Just open your jaws right there. You can push down. You don't have to go again. Yeah, so come down just a bit. Just try not to shoulder left anymore. You got a little bit of room for shoulder left. Is that all?
Perfect.
Beautiful.
Well done.
Okay. CQ5.
BioBox 2 is closing.
Box 3.
Right.
That's R02.
All right.
Now for the trickier of the samples. That was practice round.
Stamp on R02.
Timestamp is 12:37:57.
Is that when we started taking sample?
That's when we started. Yeah.
That did not take very long. It was 12:44.
Job team.
So this is going where?
High fives.
Air fives. Raise the roof.
This is going to go in another RNA later one.
Okay, so we should.
Yes,
says
yes,
one in the
please.
basket.
Yeah, but I brought the basket in because it's going to be hard to get down there.
We don't need a live sample for this one. Just RNA later, because we collected a live one yesterday.
And we could open up the RNA later right now if you want, and then I can pull the port back in and get the sample and then go.
I think having it open so that if it's a tedious thing, probably stacking it up in a folder.
There you go. Try to pull away. So that's stuff like roll that lid off instead of pull straight up.
Okay.
Just circle about top one. What's coming in? I'll give you a sit.
This will probably be pretty promptly.
I'll do very good for that. I got a good view of what's down the road.
So we can go back there.
Get you a little bit more chill. Yeah, pretty good view in the mid cam too. Pretty well lined up.
Really looks like a well, stretching now.
Rock 3.
That looked pretty strong. Taking me for a bit of a ride over here. Alright. That's a piece of the top. Do not, yeah, the top bit is crumbling. You got a good one. You want me to maybe come and stop the bottom from falling?
Yeah. Kind of want
I get a little herd of amphipods.
Oh, I see that. They do move in herds.
I will never miss an opportunity to go to Jurassic Park.
I found recently that you can buy the little shaving cream things.
Oh, no way.
Yeah, and
Oh.
I bought one, but it didn't come in time for this cruise. I was totally gonna bring it out.
Are they functional? We could use some of those.
Oh, it's like.
Not functional, but just
We could make
more,
it functional.
Like the shaving cream aspect is functional, but the keeping it
Mm.
cold aspect.
I'd pay extra for the vacuum
It
insulation.
would be a very useless nitrogen like doer, but.
Well, wow, that Alvanella just jumped ship. A pair Alvanella. That'd be good for transport. I bet
Yes.
cryo-electron microscopists would eat that up. You know,
well,
huge market there.
Or you could just pack it in your checked bag and then they won't freak out.
Exactly.
The way that they do with the dry shippers.
Yeah, that's what it'd be good for.
We've also got those bags.
This is science crab territory, but I haven't been watching.
Science crab.
Or just try to see if you can pick
Science
it up.
Crab
Like, I'm just
Junior.
here as a backup.
Do you see Science
I'm
Crab
just your
Junior?
backup.
Yeah. Whoop.
Got it. Nope.
Did you get the stump?
I got this whole track about to hit my own. You see if this is stable? That piece is stable right now. You're clear to you sit and start shouldering over.
Just gonna back up.
Okay, yeah.
So we can see what's going on.
Alright, so that you still have the little chimney that you
Can you grab it just whenever you're ready?
can grab, then on the balance. Alright, it needs to go in RNA later though, right?
Yes.
It's a little bit bigger, but maybe we can do the break apart game on the bow box.
Yeah, if we can just get it on the basket.
You're gonna take the whole chunk and put it on the bio box and
Yeah,
break it up.
That way.
It's gonna go to pieces.
Uh huh.
In a way we can't really control.
It works for us, but it works for you.
Trust me,
No, I know.
I'm making this fall right now.
Yeah.
Kind of practice.
Lots of worms.
Alright, I need you to go in there. I'm holding this stuff up right now.
Okay.
So
Yeah, I can try.
yeah, making half an inch of that. I might be able to get it. You think you got it?
It's kind of an awkward shape.
Yeah, I think if you try to go into it, see that top chimney?
I have
Get
seen
your
a
jaws in
couple
there.
of albanellid worms just kind of bail
Get your
out.
jaws just before they touch.
It looks like it has a tail, but it's really the worm's entire body. Kind of star part is its mouth parts.
In there without actually having to grab it. That makes sense? So go in like this, close, 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 go right before you touch it. Rotate.
Oh, that is a Ketognath moving through the frame right now.
Get into a bind and
It's doing the jerky little waving motions.
Yeah, and that its own weight will get into a
That's
bind
really
and
interesting to
go,
see here.
And then you can lift it.
Also known as an arrow worm. They're not actually worms. And when it comes to mouth parts, they have some seriously formidable chompers at a micro scale.
You got a good view of the nip cam. Come up a little bit. Try to lay down just a touch more. Come up with your elbow. Alright, go with a touch close. Alright. Give it a little bit of a wiggle. Yeah, okay, another. Go back to where you were. Touch close. Alright, rotate counterclockwise as you come up. Like a nice and easy touch. So you're going to want to try to rotate counterclockwise as you shoulder left. You're just trying to grip it with the jaws without crushing it.
Jack, you stay now. Sorry, it's easy to do now. I'm still on that from last night. That was not an issue for any fish up the road. I mean, if they're going to give them out, they won't look if it's on brand.
You got a piece, don't slow move. You got a
bringing
perfect
up
piece for her right now.
Your lander is still where we dropped it, which is
So you just need your shoulder up,
some odd meters on the
elbow in at the
southwest.
same time.
And I have to pick it up.
Nice
So I
and
gather, I mean,
I'm going to try to get us into
I'll
a position
bet you anything,
where
Corinne, I'll be here in exactly 18 minutes, or 11 minutes. And
I'm trying to get this away.
we'll probably be moving the lander.
Yeah,
By the time the watch.
Keep doing your shoulder up.
And that's all you have. I don't know exactly what kind
Alright, so you're right there.
it's essentially about a couple
No, no,
hours ago.
I'm frozen up.
And
arm
we
over.
should try that site that you marked up yesterday.
Alright, you ready?
Yeah, well, I was thinking grab camera,
Which you marked up the,
you know, the two chimneys with the active.
Oh.
Active 3. That's where we are right now.
Yeah, we're in active
Torch is
3.
coming out.
yeah.
I'm going to come out
Pretty
nice
sure.
and easy.
Yes.
Yes.
Have you trashed
Wait.
it?
Oh no, hang on. This isn't it. This is no. It's
This
different.
is active.
This isn't the. Oh, I'm talking. What's the one with the in the canyon?
Oh, that's
Oh, look.
That's-
I have the.
4.
I have the waypoint notes from yesterday.
shoulder
Active
up,
4.
elbow down.
So
I would right now, I would just elbow in a little bit more and then yaw to the left, get it over this front porch. As soon as you do that, you're in a way better position than we currently are. So yeah, bring that elbow in and yaw left at the same time. Yaw left some more, yaw left some more. All right, now you're over the porch, so. All right, you get to sit where you need it. All right, sits in a good position. This is gonna be another little bit big to get in. You're probably gonna have to do a little crunching when you get down in there. You're probably gonna have to
I think it'd be
rotate.
better to just do a 180 with it.
Yeah.
Dump it.
I would rotate it counterclockwise, that way you're going over that basket. But
Yep.
a little bit more elbow in to help you.
Right.
The one that's noted on here is Lander's setup one. Which we did see out yesterday. I think that's the one. Yep.
Put the coordinates.
Great.
And they're on the diagram.
Do you want to retrieve? Do you want to look at Lander setup one again, or are you content to bring Lander over? And
Is that the area we're in?
I think it's pretty close.
Don't worry about that.
There's a whole
You're
cluster here.
over it according to set.
Yes, we're about 10 meters away from Lander setup one. I'm not going to look at it before we get touched with the Lander drop it. I just need to make sure that Pharaoh's happy with the possibility.
Now you
I sort of
can
thought
develop
that they
again.
weren't happy with it yesterday.
Yeah.
Tap it in. Tap it in.
Grasp.
High level camping.
I'm pretty sure that alvinellid worms were the inspiration for the monster in Stranger Things. I'm pretty certain of that. Star-shaped mouth parts flare out
Freeze.
somewhere between that and a kitty math.
When you push down that hard, especially with something like this, when you're pushing down, you want to do the freeze every now and again, just so you don't have that much pressure. When you go and hit it, it just goes insane. So freeze, do it again, freeze, do it again. The piece that's hanging out more forward with that vehicle, if you were to kind of get that to
7-to-1 ratio of RNA later to sample.
I'm not positive about it, that's just a tactic. Trying to break that forward piece on.
I mean, 1 to 7.
No, further away from the camera.
No RNA.
Pushing down over
Oh,
there, trying to
exactly.
get it to track
Exactly.
right there, opposed to the stuff we
We understand each other.
Yeah, like that area, I'll push down there.
I kind of slacked up on the filtering effort, by the way. You probably noticed.
You slap.
Well, I didn't put it back on or anything last night.
Oh.
After they finished up with the pump.
That's
Still
fine.
poking out a bit. They're gonna have so much fun getting that out of the
I just want to say that it's so tight in there now already. It's not gonna come out.
And we have a rubber mallet in the lab. Let's take that.
We have technology.
We need a hacksaw and just
Okay, I think you're good for lid the rest of the way.
Yeah. Does anyone want
Nicely
some
done, man. Good.
RNA later?
Alright, shoulder right a little bit.
Pickled worms?
Yeah, we might have to borrow. Maybe, yeah. I might have to borrow that big tubing cutter from the geologists.
We may have just donated a coral quiver.
No, I have a modular
We'll
one.
just bring home the coral quiver.
What are we doing after he secures this
After he
lid?
secures that lid, we are gonna pop over just to lander setup 1
Okay,
and scout the potential lander position there again before we go to get the lander itself.
roger that.
I just want to verify, do we have any other
No.
geology or bio objectives here?
Yeah, be careful.
I don't think we have a slurp here.
Use your yaw function a little bit more when you come in.
Animals slurp on a high T chimney.
But I think
Yes, I mean,
on
you're still
a-
clear of it, but it's just
Height,
good
a bigger
practice,
high
you know?
temperature.
I think that they're wanting to
It leaves you
do
with more room to be able to use that instead of being
another, like
up
when,
in this
well,
height.
Yeah,
after,
in the
after
F.
IGTs happen.
Great, at the F complex, which I think is
Correct.
hotter. Yeah,
Words are hard.
you're doing great.
Yeah, and then
You can grab the eye too if you want.
There you go. Gorgeous. Right up in
That way
there.
it's so good. You can just get it to see that it's hooked on one side and then just.
No kidding, do we have someone watching from Galapagos? Welcome.
Yeah, it's probably gonna want to pop this side off. I think the way that you were going was really probably the best. You can, the only problem is when you come out there, you gotta be real gentle because if you get hung up on it, on these back little flanges on the heart, you pull it off and you get very angry at
building
yourself.
anticipation and trying to get speculating. Just saying.
That's it, I mean, like even if you would've got it in there, like trying to get out of that situation. So like right there, rotate your jaws a little bit, clockwise. Yeah, like that, and then push down, try to come over it as you're pushing down, so.
Filming future
Just extricate myself at this point.
Yeah, if you try to just come straight down on it, then. You can also use your jaws closed and push down on the eye. That's also another way to look sometimes for getting that center push. Yeah. Keep it from rocking, but that's, it slips off the line. If you can get that right side up for it. See that little nipple in the T4, that little indent? Push it back, pull it down a little bit, kind of locks. Yeah. There you go.
Nice. Well done.
It's in the hole.
Fantastic. It was nice to see the RNA later coming out there.
Yep.
He did just 35 minutes.
All right.
We're just gonna push 5 down a little bit more. It looks
Okay.
like we might
Great.
have
Uh-huh.
opened it up a touch.
Huh.
And then I'm going out of this hole for pick it up the rest of
Bienvenidos a nuestra misión segunda en el campo de eventos Pinguinos.
the same thing.
We have just finished our geological sampling for now.
Shoulder right touch.
The watch will turn over soon. I'll bet the new lead will show up in exactly 6 minutes. And
Corey
then,
will let me know.
yeah, exactly, the lead. I'll show up in
One
6
thing we have
minutes.
to do as well is tighten these up. These are a little-
And then we're going to get on with our lander operations.
You want
Thanks for joining us.
tight on there before I sort it out. Alright, you can go first though. I'm gonna take off and I'll need you to stow the port arm. This is how we used to have to operate all the time before we had Rock'em and Sock'em. You'd have to get one T4 out there to just like do that free solo, like 2-finger grab kind of thing.
You had one
That
minute,
grip.
or you had 1 minute, or you had a
No.
precision minute on one side.
No, before we had Rock'em and Sock'em,
Mm.
like to get in pairs positions like
Oh.
that. This is what we would have to do, put one arm out to like get ourselves stable
Hmm.
and then go do sampling.
Are those the names for the rams?
Rock'em and Sock'em.
All right, Roger.
Talking about commercial jingles.
We are here. What's that? With the portal? No, I'm gonna take off first.
Still under 15 seconds.
We are gigantic children.
I don't think that's a toy that could sell today.
What are you talking
They have
about?
something out
I have one at home
Yeah, I think
for my boys.
they sell them.
Oh, they still sell them?
Oh yeah.
All right, we're gonna head to lander setup 1.
Lander setup 1.
Just as a reminder, we'll see how this plays out, but the tracking appears to be drifted about 5 meters southwest of where it was yesterday. Based on the visual fix on this chimney.
Gotcha. The ROV tracking.
The RV tracking. But you know, it's based on one fix, so.
Lander setup 1. That's just directly in front of me. Lander setup 1.
I think that this is
This is
it. This canyon here. No,
Yes.
the lander setup 1 is on
The other side.
Yeah.
this side of it.
But there is venting.
the right hand side of it.
There's
And
venting.
the proposal, as I recall, well, there's a fish in there.
Fish.
There's a arsid by the looks of it.
Is it a zoarcid?
I can only see the rear end.
That's a fat one.
Big
You
bone.
want to label it fat zoarsid?
Fat zoarcid.
Fat shaming.
That just means successful. Hey, there are no judgments here. It is an observation. Yeah,
Look,
I'll let
it
Dan.
is a shame.
I'll let Dan take the reins here.
Oh, world of power.
Yep. Imperious.
What would be useful is just to have a little. One of the benefits of this, I think, is that there's a bit of space to move around these two units, right? As opposed to it's like we attempted yesterday where we were crashing every goddamn thing. This one I think has a bit of space around it, but its priority is finding a good spot, level spot in the ground bit further away from it. So maybe just
So like just
Level or
fly
just
around
smooth,
and
to
then
clarify?
how
smooth.
and how
Okay.
Longer it goes.
and how
Understood, different from
how
level.
far
though.
away are you
It's
hoping to
on
be
the
from
left
the
side, yeah.
That's so weird.
See how hollow like the chimney where it's hollow right?
Yeah.
Do you want more?
Yeah.
You
my,
want the
my,
lights
my
on this
sense
side
of
or this
distance is a little.
Right.
There's a little.
So kind of
That's
like what
relative, but
kind of
like-
like what we did at the Chimlet. You want that
Yes,
kind of light in
that
front?
kind of side, that kind of
And
space.
do you want the lights on this side or the lights on that back side?
I think it depends on where we have a spot.
We haven't been lasering anything.
If you
Good thing
come back
we've only
from
been
this
looking at
side,
things we already
there
saw.
was-
Yeah.
There's a
Some
chimney.
pillow.
There's a
Some
chimney
pillow.
to my back, to my port side. But no, there's nothing else to my starboard. Well, I don't know. We'll see. I'll have a little
Where's your
spin.
rear facing camera? Is it that one down?
I'll have a quick spin right around. Oh, there's another one over there. But it's kind of an optical illusion the way it is. No, this is definitely not going to be a good effect. It makes things seem closer than they are. They're there,
Right, I see.
but they are still close. So, without putting the lander like legitimately on that thing,
That's just
I mean,
Which
which we
would
want to
be
go far enough
cool.
away that we'll get that little.
so I mean, there is this little thing right here, this broken
That might
chimney
be
thing.
too close. I don't know. How far away from you
Hmm.
that?
It looks like,
It's, I don't know, maybe like 10 meters away.
That's 10 meters away
No.
from nature.
yeah, it's drifted.
No, I really don't understand distance.
So I mean, we can maybe set it on top of that if you're okay with that.
Are
Like
you
with
looking?
this big rock.
It would be-
setting it on there.
invert.
Yeah.
Would be strange. Yeah.
Yeah.
Some of them are crunchy.
But
But I really like the squishy ones.
So there's just a massive load of folders.
708.
On the other side.
Look at Corey.
I like
What?
to my
The trains and the watch leads run on time.
Sorry.
Like clockwork.
I promise I don't check the time when I come here.
Is it worth just doing a
It just like works out like this.
We love it, Corey. Okay.
My routine, getting my tea and everything.
Yeah, I definitely I want to get a what's going on here before you start making me turn the lights off. So this area over here.
Here, you get the punctuality award.
We'd be disappointed if you weren't here at 7.
We've been talking about it for the past 40 minutes.
I mean, I don't think that's the direction you're going to want the light going. Looking behind me, like, yeah. I mean, that's pretty much what
So we're re-scouting the lander setup right now. And the
Let me
lander
look back at the chimney.
is a good 150 meters to our southwest.
Range on the
We haven't-
We haven't really hit CO2 for the lander
There's
is like
5 meter range.
we haven't even played that as yet.
So
Turn around,
So that's the
have a look at
next
those.
thing on the
Next up,
plan, the
once they find a spot.
filming. Yeah.
Yeah, there's a possibility if they can't find a spot here that you'll have to set up the lander on the F complex, waypoint F, or elsewhere. That would be Smoker 1, waypoint F, but I think that's where they tried finding yesterday.
Big chimney.
After that,
Let me come up a little bit higher.
so objectives that have been
Little
met
Marcus heading in.
are super,
Forward.
Yeah.
some live sulfide objectives.
Where my
I
life
was
is
so
gonna
happy
turn
something
into.
when I
Actually don't have this all up to date. We also got a little Benthics F104, which was fun, first thing.
Oh, I
in the
saw that
section
in
sampler.
the overview. Yeah, that's
Side
Um,
onto that chimney.
Yeah, we're pretty much side on.
We have not gotten a background super yet.
No,
So I think
that
we want
can
to
happen at any time.
Yep, so if you have some spare time.
Yeah.
But it can't be near a chimney.
Yeah.
An animal super, which is actually 2 filters.
Oh, and okay.
Otherwise, it's RNA later only. This is the main thing you need to know, is that once we're done with the lander ops, or if there's some major hang-up in the lander ops, you can go to F complex, which is not, well, it's not super close to where we are, but it's also not super far. It's under 100 meters.
Yeah, but I was wondering if the IGTs are probably have to be done
Well, we have
relatively soon because they have
to send
2
them
of
up.
them have to be.
Correct. So I think the thing will be filming then before you interface with the lander, the IGTs immediately after Dan's done. Wake Jill down here. IGTs 1 and 2. Send them up.
Where do the big ones go?
And I mean, that'll. There's 2 hours of filming allocated, so.
You can also check off this piece.
Cute.
So filming, and then
That's all very steep on this.
Jill is hopefully maybe up already, otherwise waking her for the for the first 2 IGTs.
That's right.
And then, because they still have to,
That has a nice
we haven't taken any IGTs yet, correct?
We've not taken any IGTs
Yeah,
yet.
so they have to be,
Accurate.
yeah, they have to be taken and then on the lander release.
And we've been, so you know, they're on a, Dan's crew's on a 2-hour clock, and I'd say we're about 12 minutes in. Started.
I think these changes
Oh,
okay.
right?
Yes.
And you can read if they're having a really good shoot, they're having a really good shoot.
Yeah, no,
Um,
if it's great, and then we can give them more time.
Started about 7 o'clock.
Okay, sounds great.
Anything else?
No, it's pretty straightforward.
The only other thing with the super samples is that, like, you'll do the parallel temperature wand with super sample, and then you'll put the temperature wand away at the end of the sample. Hold the super wand there. Pull out the pH probe. Bring it to the super wand. Hold it for 5 minutes just to get a pH reading.
Okay.
It's written down.
All right. Oh, it's still good to know.
Did you get any of these? That was after you.
Yeah, I put it on my bottle there. This is my bottle. I'm so happy for you that you found this yesterday. It's like unbelievable.
I have more.
The backside doesn't look bad either. This is the backside.
Now.
Which also looks nice as well.
Yeah. I just have a look at the terrain. Maybe just come down and then back off and see what we've got in terms of blank spots, or even slightly angled spots. But we remember what the camera angled at this time.
You're good friends to historical.
Oh, that looks like a good spot right there. Look at this big old rock. Not that one.
Did we see any
Right here.
elements yet?
I don't know.
Which I mean,
No,
like,
so
that's
you're-
a
Just elements.
Yeah, and we were at active 3, so it
I don't know.
was pretty quiet.
I think we've observed mussels. There's some small mussels on active 3, but like nothing
Like,
collectible.
yeah,
anyway.
mostly
Set it right
seagrass,
there,
no
that's not
like
a
big
problem.
colonies.
Yeah.
You said it.
Yeah, this would be very interesting because
Yeah,
this
it's gonna be
How
strong.
big did you get?
Seems very,
How big that is?
Yeah, it's just
That's
really
narrower
fast.
than the seat
Yeah.
of your lander actually.
Amy made a good point yesterday too about like possibly the metal concentration.
How
They're
far away
higher
from the
gel or
chimneys are we
something.
now? Sorry, at least, you know.
No, I'm just
Yeah,
curious.
let's try that over that.
So it's like toxic for a lot of the
That's
animals.
the metal.
Things
About the metal
tend to get stuck behind
sulfide
you.
ratio.
Yeah, I did not, like
That's the
yesterday when
center.
it was noted, I did not realize.
Yeah, that's the center.
So yeah, it would be
So,
interesting to see
like,
to get
we
the
keep it
chemistry
in line
data
with
and
the
some
rock.
of
It's
the
shooting
information about
at the
that.
center of the 2 that you want.
Thank
Can
you.
drop a marker here just so that we know this is exactly where we want to come back to, or is it would you be able to find that relatively easily?
No, I would like to put a mark
Spot
there.
people.
Because there's also another rock right behind me. A lot of things are falling down here. Just in front of me, yes.
And this is something that can pretty.
Oh, this is Jacob's book.
Although, I mean, I guess.
Is there a spot further back, or are we getting too far away?
Ian's
I
last
would
full
think you'd be
limpid
getting too
list.
far away,
It's
but
very funny
I
too.
can also still check that out. Lander.
And drop side.
So there's another rock right below me right here that could work. This is just all big chimneys that are falling over.
I wonder what those dangly things are.
I was gonna ask the same thing.
They look
They almost-
vaguely reminiscent. Go ahead.
Oh, I was gonna say maybe iron oxidizing
That
bacteria.
one does,
Oh.
this is
That
further
was
away,
more hopeful. I thought
another
that
rock
might be
that we
the
could
key,
straddle
yes.
right there. But then it starts
Probably gets
dropping off
bacteria.
quite a bit.
Starts dropping. Side bit.
Yeah. Like South Florida.
What do you think this side's
harder than the other side, or?
No, this side's easier because there's not as much power.
Behind
Yeah. Although, I mean, this side will be easier to fly on.
Yeah. The hardest position around there.
Yeah, so this side, there's not as much for me to run in to
Yeah.
be flying dark. The other side, there's significantly more of that.
Okay. So in that sense, we want to be on the other side because this is, so when we'll be flying dark, that will be when random lights are on the other side.
Correct.
So we want to
So
land on the other
this
side.
looks really clear for me to be able to
Okay.
fly
So
around in the darkness and not destroy everything.
if you look on the other side then, let's have a look, see if there's somewhere else on the other side.
So, and that depends on, because you wouldn't want any of my lights on, right? Kind of like last time?
Yeah.
But would I be able to have my half light on?
Yeah.
Okay, then I'd probably be okay on that other side, but this side is super.
I'm happy to film from this side with the ROV, but it's just whether we can get the lander
On that side.
position on the other
Yeah,
side.
the F complex were the larger events.
Yeah.
Going in, the larger events
Yeah.
we visited yesterday
Light it up.
that were described from a previous expedition,
The
I
thing is,
think.
those lights are very
That's where that letter came from.
they cast a broad light.
Though I think on
right?
that
So
chart
you
it's
can
E.
film from a higher altitude.
I think she
Or is it
corrected
G
this
or-
yesterday.
Using the
Oh,
ROV,
it's
but it's
good.
more
Um,
on the other side.
this
you'll
was
be able to
corrected
see the
yesterday. I think
landing.
all of these should be correct.
They're going to be able to see the ROV. working.
Yeah, we kind of were like totally
The
confused.
ROV
And it's like,
will have the
can
lights
you
on.
redo them?
Am I making any sense?
Yeah.
Some sense.
Some, yes.
Yeah, and this was basically not there anymore, right?
Sorry?
This one wasn't.
Yeah, that was the roll up clamps there.
Sorry, I got a lot of lights on here.
Interesting. Maybe we don't find this really anymore.
Let's go out this direction right here. Straight out right here. And see if there's-
Okay. At this point.
At this
It will hit me about halfway through breakfast.
Oh, God.
So,
Right now I have enough energy
I mean,
to go to bed.
I think we can land it here. I mean, it might not be, it
It
might not be sitting
Well,
with
you
all
also have-
four legs down, but I mean,
That's
we could
true.
also still get it off high
Yeah.
to get the.
What about like in here? Is that-
Yeah, I think all that's doable.
That's
It's not,
why
I don't think it's gonna
That's why you
slide
were
down
good.
as long as you got feet on the ground. I mean, it
Yeah,
looks like
probably
it's 2
like
feet
700
are
calories.
touching.
Do this way.
Breakfast and go.
Yeah. But, I mean, it's not gonna just go tumbling down.
No.
It's just, I mean, it's a lander. It's,
Yeah. Worst case, it's just going to be leaning.
yeah, or if it does, if it slides, it's gonna stay upright.
I need to mess
Yeah,
with
fine.
It's not top heavy and into that. Complete opposite of that.
Okay.
So, I think this is a good spot. So.
Spin round.
Tomorrow, to be careful, it kind of
Down there.
a few minutes
It
later.
feels like a bird.
Oh,
No,
like,
just you-
yeah.
You don't try to force it, just, we just wanna see your cadence. Oh, there's a T for, Jacob, bye,
Bye.
2-4.
He doesn't like those ones.
He doesn't like these
No,
ones?
he likes them. He's
But
just-
they're not
that's
They're not impressive.
Not
that's
as
not-
impressive.
Yeah, that's not
Got
on
it.
his list.
Okay.
It's not his hit list.
I
So
think it's
behind me
pretty.
right
I
here.
like it. You're,
Now, is that the two?
you're amazing
Yeah,
just
those
the
are
way
them,
you
right?
are.
Yeah.
Yeah, that's it
Another
right
one from
there.
yesterday.
That white spot's the
It
pretty
was
positive
very
the
pretty.
center of what
Wow.
you're
Yeah.
Yeah.
So,
Did you
like
get to
here,
see it
I'm just
light
gonna
up?
sit down, I sit
I
down the
didn't
same
know.
way as the lander.
I
pretty
still
much,
do,
in
huh?
through.
I didn't either.
So,
And I just saw it when it came out and wanted to have a quick look at it because I had Nicole with the samples and I was like, okay, yeah, let's get that done. So
yeah, I mean, that's
she doesn't have to do
me
it like,
sitting down.
Yeah. Yeah, she stayed up pretty late. Yeah, 'cause
Spot.
I went to bed at like
Okay, well, let's give that a go.
Where's the
9
lander at?
a.m. and she was
You're
still
asking me,
up.
right?
oh no.
Yeah.
Is that there?
Maybe I thought we would.
No, we put the
lander dumps, that's just where we.
Or you leave
Well, I
left
think
it?
she was taking photos.
Or you
That's where we just came from.
Right.
Little guys.
She said we were safe for 2 hours.
I don't think that's on that, look at it.
No, yeah,
we
Yeah.
have right in the south. See that blue line?
Yeah.
That is BBC
It's
lander.
way over there.
Ah,
Are we sure? Can you verify that on the Ranger, before we make that move? No more calls. What am I gonna do with my life?
You missed. Yeah, what's it?
What kind?
One that you don't like.
Lobate? Are there some for?
Oh,
Are there
that's
some that you
interesting.
don't like?
I love lobates, but you can't suction them. Most of them.
Why can't you suction?
They come apart.
Oh.
Depends on the species. But
Challenge
Where's
accepted.
your- oh, yeah.
Okay.
Yeah, yeah.
I heard that, Jared.
Yeah,
Yeah.
what?
It's a give on it, too.
What did he say?
Yeah.
Is it challenge
Challenge
accepted?
Yeah, we think that's.
Oh.
He's gonna go after everything.
Was it, what, did it have any color?
They had a red
Double check
gut.
this.
Oh, that's a Cyrobi.
So
Yes.
this, the chimney right here.
Yeah, I'll buy a J-Rod a beer in the mess. Get that one piece.
Well, we only have non-alcoholic beer.
Right now.
I'll drive us out. Good part. Thanks.
You're the real MVP.
I actually did think of
That
I was
one
like,
over there, so.
So,
This.
Those are
This one-
fascinating.
I was looking at the wrong one.
this would be the most wanted of all.
Honestly,
I think I.
well,
can
we'd
find
actually
it.
hit both of the secondary most wanted.
So
So
you wanna be further away?
we got,
from your
I mean,
stuff?
we
yeah,
got
this
this,
is pretty
this
good,
one
actually.
at the beginning of the dive,
This is fine.
Yeah.
which the colors represent a turbine set on.
Yeah, we moved that, just drag that
Oh, yeah.
And
site two to right.
we got the red one. Yeah, this one or these ones
Yeah,
are
throw 'em
going to be
there.
hardest to find. Totally undescribed. They're benthic. They're nondescript. They're just like little yellowish blobs. They spotted them on the EPR.
How big are they?
This-
2 to 5
are we
centimeters.
talking about dandelions?
Benthic ctenophores, not dandelions.
Oh.
Benthic ctenophores, not saprophytes.
Oh, those are
And take eagle eyes.
Those are the white things with a big open mouth? They kinda look like a
usually
ship.
Usually the mouth is actually attached to the rock, but they got two tentacles. The telltale is these two tentacles. These are from what dive are these photos from? 543.
Alright.
Yeah.
Wow.
It's
Okay.
kind of more flat than.
Yeah, it doesn't look too bad here.
Yeah, that would be number one.
This one,
Okay.
and then the other one is
The other one is the giant blue one, which
Yeah,
you can't
do that.
miss. I mean, like if you see one,
Do that.
Let's talk
you
about
will
the
spot
moon.
it.
How would you take lights on there?
Yeah, it's gonna take a
My
little
favorite
bit.
animal.
We have some time.
Yeah, you can put it and then measure and be like
First dive during the
75 meters away from it.
I guess my point being, if I put it a little north of it, then you can run out the stern of the ship and not have to move the ship quite as far.
First dive, I think was
That works for you.
263?
It does.
No,
It
262
does
Here.
indeed.
So
No,
that's about
no.
85,
Overall.
87.
Overall.
262 was first dive, 263. It's, they originally told us the wrong.
We're at the ROV.
You mean 562?
Yeah, sorry.
go ahead.
Forgot.
Got an updated waypoint for you.
Yeah, they were just at 562.
Slice away.
Thank you. I keep going to every single time.
Nice t-shirt, Corey.
Thanks.
I have a blue one, but I didn't bring it. You're gonna get one each or
Yeah, I
something.
only got the
I like the
gray
blue one.
for some.
The deep sea bio.
It's
It's
no,
so
oh,
Fire
Mari.
resist. What color Lance is now.
bio.
You're going to pick up the laser.
That is correct.
Cool. I will observe that. It's been logged.
I should actually put it on your shoulders. I did play around and see what I did this several years ago.
It's pretty straightforward.
Are there any particular annotations that are hiding in places I wouldn't expect to find them? Is everything under science?
Everything is under science.
Everything. Okay.
Yeah.
Including lander stuff.
No, that's not science.
Okay. Or I guess there's all
Whatever, we put IGTs on it. Got it.
IGTs render fluid. I think the only thing that's confusing is for the super. So you want to do super start at the time of the purge, and then you'll note in the description the time that you actually start pumping the sample, and then super ends at the end of that.
Okay. And there's does anything need to be added in the
You only
description
see
at the
one.
end of that?
Yes, so you
Then you could
want
be referring to multiple.
you want
Last
well, I
I
guess
counted, I think
that's
there were
all.
5.
What, temperature,
Temps,
coordinates?
yeah.
Oh, the
Yeah.
coordinates are in there already.
Coordinates are in there, depths in there, but yeah, you'll want to do the temperature range, and then also note when the
Yeah,
pH.
the white ones are the easiest to see.
Logger
And
is
when they're
out.
red and blue, that's when they get a little trickier.
Oh, yeah, there's a red one. Is that the monitor or the camera?
Start.
Did Alex get some shots of her? Do you know? Yesterday?
Sorry.
Did Alex get some
Alex got
shots?
some. I got some. I have some dark field images that I'll share. My camera started working.
You're right, once you see them, there's no one seeing.
I would love to see those.
Yeah, I'll share them around. I have no idea when I'll find time to
Yeah,
edit them, but
no, it's that's all right.
I was looking through Alex's photos. He made those birds look very yesterday.
Yeah.
Right now, you have a track.
We did.
It's not using that one.
Well, it's also bigger.
I
Yeah.
got a Tina Ford question. Which one is this? This was from 260. Or
I saw that one.
Yeah.
That's a really good question. I think it's about Ecuador. Ecuador is really
some sort of
basic snowflakes. And I'm sorry that they don't have, I think when I give them a common name, I
Once
just
you
go
come
with
up
the
with
deep
get
sea
rid of the
location,
lasers.
the ATHO means to keep.
Did you text
I totally
me?
understand what
I
you're
haven't
saying.
messaged them yet. As soon as I have them passable photos, and I have data, I have all the stuff they want.
Good shots.
Yeah.
They do like highlight videos.
I was just curious, because
Oh,
I was
yeah.
saying, I was like, who the heck is this?
No, that's about the same. I wouldn't expect the annotated that way. The annotated look big. Yeah, they have those big, like, sometimes they look Mickey Mouse style,
Probably
they
they're
look
not
sort of inflated.
Yeah.
But sometimes they look kind of like a TIE fighter, where they have the loads are flat and outside, that they have, I guess, lowers the point of page.
Okay.
If it's like a
Yeah, but you
football,
could almost like just say like,
it's a site thickness color
Okay.
harder
That's
to
a
remember.
big anemone.
It's a very specific way to
That is true.
It's named after, I think the site was a water nymph,
Oh, okay.
so I don't know if that helps.
No, that does.
People pictures.
No, that
Um,
does. It really does. Okay, cool.
Yeah, there's only three that I know of one described species of Baphosaurus, and it's from like the north.
Depending on how far away it is, they actually like turn.
Okay.
But we know that there's like from genetics and morphology, they're just everywhere.
Okay.
That's a body schema.
That's crazy.
Funky guy.
Yeah. Yeah, the cool thing, the thing that I find really interesting about Baphosaurus is there's Baphosaurus and there's this other type of lobate called Bolenopsis that we've also seen in these dives, and their ranges overlap by like 2,000 meters.
Oh, okay.
The Bolenopsis, they both have really wide range, wider than most ctenophores. Bolenopsis goes from maybe like 1,500, 2,000 meters all the way to the surface. Like you can catch them snorkeling on a good day. Baphosaurus goes from like 2,000 to 3,000 meters up to about 200, and then they stop. You never see one on the surface.
So, we can do background.
Well, I mean, I have a
and then
hypothesis
hold on.
as to why.
Like a big patch of
Right.
animals.
But,
Yeah. Yeah.
it's sort of weird how they evolve that way. Like they both handle the same.
I wrote
you
it
know,
down here. It's like
absolute
one thing to keep in
difference,
mind.
but
For every
not
super
at the same
sample,
place.
the wand should be parallel with the sampler.
Oh.
Yeah, and then
Sorry, yes,
when the sample is
No, I like
taken,
it. I like it.
change out the temperature wand for the pH probe
Yes.
and just hold it there for 5 minutes.
Yeah.
But yeah, other than that, we also slurped
Crunch
the
and
dandelion
mimosa.
siphonophore.
Bloody Mary.
Okay with that.
Yeah, we got some
Apparently we can't have them in the
sulfides
national
and
park.
pearl critters, so I think mostly we're on the animals and geology and IGTs.
Sounds horrible.
It does.
Going up. Going up. Going up. Enjoy breakfast.
Sunday.
Sunday.
myself for these.
No, that,
Or I
do
forget,
that.
or I lose my thread. So we're just done. I'll see you up there.
That is correct, sir.
Um,
Scooting on over. How's your finger?
Damaged. So
Yeah.
the cups, but it's alright.
Yeah.
Thank you for asking.
Yeah, you're
me.
here.
Happens once
I
in a
get you.
while. In this line of work.
So did you find a setup in that area after all? Did you find a setup?
Yeah. Well,
Give it a
possibly.
shot.
Possibly. Is
There's some options.
Dan is coming back on in a bit. Yeah.
Mm-hmm.
keeping the delta low for.
Just got a little strung out and it's flying fine. I normally don't have you start paying out until I start when you're porpoising, which gives me an idea of what that break point is. It's a good way to, because when you change, like, you do me a favor, go to your sensors. So you can see this. So they go to dials over here at the same time. And then so go to CTD and watch your delta reading. Delta 38. Yes.
Oh boy.
Okay. So now hit UDSGX. So there's always going to be a discrepancy in depth sensors. There's something like it to be a scientific
Gladly
depth
obliged. I
sensor.
mean, when I know what it is.
There's like a 1%
It's like, what's that? It's pretty inflated.
deviation that's allowed to have, that's acceptable.
Yeah.
1%.
How long will you have
So when you get down to 2,000 meters, so
Probably like.
although our
That's fine. It's
NTs
better
are
than
going to
one.
have them all the same.
More between
and
all
they're
three.
doing
Kind of-
correct.
Yeah, the more they can pressure for
Yeah, and so they make the
I like
adjustment for
your
that.
process of
For us, you could be sitting there showing 39, but
It's actually 60 or whatever.
We do all those things we did the other day, we just think our vision system has like. ROV, remember?
Oh, the fuel measure the power source?
Well, it's your power source.
Oh, the
Yeah,
oh,
like,
the
yeah, like they go away from the direction of the camera, they come back in.
Scientists
do
do
that.
years.
And then
Other
go
junior.
Oh, yeah.
Might as well.
Yeah,
One
it's like they like to know it.
Not as well.
Not as well.
Not as well, it doesn't sound right.
The most
Oh, extreme.
specific and sometimes wrong. Like,
Alright,
Right.
so coming from behind it.
even if you just annotate it, those make it.
That's better
better.
than trying to go
Yeah.
all the way down.
Right.
Yeah, that makes sense. Remember that
Yeah.
was, when we were doing oral deep sea dive, that was the big thing, they're like, don't just backpack. Just don't even try. It's most effective.
I've heard corals are up there. They're microscopic, they're genetic,
Yeah.
super
You
cryptic
won't know unless
species.
you can take a sample and it's such a fish. Unless it were very, very distinct. We're like, yeah, nothing happens, so listen to it.
Where did you do some corals?
We did, I was, off the Lake Plateau, Coral Mountains, deep sea Coral Mountains, on the Lake Plateau, near South Carolina. So, the Yogi Campus Explorer did a bunch of dives down there, and Advisor was a part of that, and I helped with the content mapping, and, it's sort of like species diversity, like I don't have a ton of that, but I still did that, I think that was GE poster. Research, as an undergrad thing. I would love to do more of that.
Well, I feel like math is really in demand. Like
It
you can almost
is.
work with any discipline you want.
Yeah.
It is about the ocean.
I'm needed somewhere. I really like that. We're doing a lot of water column stuff now, so there's
Who is?
that because I have the water column just have that data and I'm very interested in it. So we had well, just as we were coming in to port, earlier before the dive started, we salt, either bubbles or fish or something.
Oh, okay. We're talking about like ETA type.
Yeah.
Yeah.
Just water quality. And using that types of things.
Yeah.
It's been very fun and interesting.
I have some friends who are really, who are really good at it.
Yeah, it's it can get very, very particular. Yeah.
I was on a Rhode cruise
Oh yeah.
a couple of years ago where we were doing day-night trawls, like trying to catch the scattering layer in their sky. Like when it was down or when it was up. And so, everybody was going to be waiting on deck and then Yvonne would like the either or something. We had a couple of fisheries type sonars and you would like point to some random station. Get on the deck.
I got one line.
I didn't know it's the yellow one, the red one.
It's amazing.
Target 1,200 meters. Target 1,260